kandungan gizi yang tinggi sehingga diminati oleh masyarakat. Daging ayam memiliki kandungan gizi berupa protein, sodium, potasium,
Baik,
vitamin
peserta
A,
masuk ya
vitamin
teman-teman.
B6,
Siap-siap, ready.
zat besi, kobalamin, magnesium, kalsium, lemak, dan kolesterol dengan total 239 kalori per 100 gram daging ayam. Ayam yang beredar di Indonesia dibagi menjadi 3 jenis, yaitu ayam pedaging, ayam petelur, dan ayam hias. Yang biasa dipasarkan untuk konsumsi merupakan jenis ayam pedaging. Berdasarkan data dari Badan Pusat Statistik, produksi daging ayam pedaging pada tahun 2017 mencapai 2,14 juta ton, meningkat 4,75% dari tahun 2016 yang hanya 2,404 ton. Namun, tahukah sobat, saat ini 80% produksi ayam dalam negeri masih menggunakan ayam ras, di mana untuk bibit dan pakannya 100% impor. Hal ini dikarenakan kondisi ayam lokal yang dianggap memiliki daya saing rendah, tidak efisien, dan memiliki pertumbuhan yang lambat. Melihat kondisi tersebut, IPB University berkomitmen untuk menciptakan rumpun baru ayam lokal yang mampu menggeser dominasi ayam ras, sehingga negara dapat mengurangi beban impor bibit dan pakan ayam, juga memproduksi ayam ras secara mandiri. Rumpun tersebut diberi nama rumpun ayam IPB D1. Ayam IPB D1 merupakan ayam komposit lokal yang telah dikembangkan sejak tahun 2012. Dengan komposisi genetik adalah 75% genetik ayam lokal dan 25% genetik ayam ras pedaging.
Jadi latar belakang dari pembentukan ayam IPBD1 ini mengingat ayam lokal ini kan peranannya secara nasional masih sangat kecil. Ya. Jadi konsumsi daging ayam lokal itu baru sekitar 11% sampai 16%. Nah ini karena keterbatasan bibit ayam lokal. Ya. Nah, jadi IPB mengembangkan ayam IPBD1 ini untuk khusus ke arah ayam pedaging lokal unggul. Ya. Tumbuh cepat, kemudian dia lebih tahan penyakit. Jadi bisa dikembangkan di pedesaan dengan pakan lokal ya dan manajemen sederhana di pedesaan.
Rumpun ayam IPBD1 adalah komposit hasil persilangan antara jantan F1 dari ras pelung sentul dan betina F1 dari ras kampung persilangan, ras pedaging peran stokop atau kampung meat type yang menghasilkan keturunan J1 PSKM. Untuk selanjutnya dikawinkan secara inter sampai generasi kelima. Ayam IPBD1 memiliki waktu pertumbuhan yang cepat dan mencapai bobot potong pada umur 10 hingga 12 minggu.
Nah, jadi keunggulan dari ayam IPBD1 ini bisa dipotong pada umur 10 minggu. Jadi sangat cepat pertumbuhannya. Nah, apalagi kalau pakannya sangat bagus ya. Nah, ini kita kembangkan karena targetnya untuk masyarakat di pedesaan, jadi kita targetkan proteinnya sekitar 17% ya. Nah, dengan protein 17% aja umur 10 minggu itu sudah bisa dipotong. Jadi jantannya sekitar 1,3 kg, betinanya sekitar 1,1 kg. Nah, jadi tumbuh cepat dengan pakan yang protein 17% tadi. Nah, kemudian keunggulan lain, karena ini ayam komposit ya, jadi kemungkinan bisa kombinasi dari beberapa gen dari 4 rumpun ayam tadi, ini menghasilkan daya tahan yang lebih baik, terutama terhadap ende ya.
Pada akhir 2016, rumpun ayam IPBD1 telah diuji cobakan di kelompok tani Sinar Harapan yang berlokasi di Desa Panumbang, Kecamatan Jampang Tengah, Kabupaten Sukabumi. Dan kini telah mencapai generasi kelima. Uji coba ini dilakukan untuk mengetahui daya adaptasi ayam di kondisi pedesaan, dan hasilnya adalah ayam IPBD 1 dinilai memiliki resistensi yang signifikan terhadap penyakit Newcastle disease atau biasa disebut dengan tetelo. Pertumbuhannya pun lebih cepat jika dibandingkan dengan ayam lokal setempat.
Nah, yang di IPB ini kita arahkan ke arah penelitian. Nah, dari hasil penelitian kita bisa mengembangkan 2 galur ya. Jadi galur yang ke arah ketahanan penyakit kita sebut IPBD2 dengan indikator imunoglobulin Y-nya lebih dari 10, kemudian titer terhadap ketahanan terhadap ND-nya di atas 3. Dengan harapan ini nanti ayam ini bisa dikembangkan dengan ayam organik. Ini mungkin bisa dikembangkan di pedesaan juga. Nah, tetapi yang satu lagi, galur IPBD 3, ini ke arah kecepatan tumbuh. Nah, ini mungkin perlu pakan yang lebih baik ya. Nah, sehingga ini bisa dikembangkan ke arah jantan pedaging untuk di industri pada umur 10 minggu bisa sampai 1,6 kilo. Nah, mudah-mudahan ke depan ini bisa menjadi mengembangkan perekonomian pedesaan.
Diharapkan pengembangan ayam IPBD 1 dapat meningkatkan ketahanan pangan sumber protein hewani asal ayam lokal, diikuti oleh semua komponen agribisnisnya, baik berupa bibit, pakan, dan teknologi budidaya, penanganan produk sampai pengolahan dapat diakses secara mandiri. Jika itu dapat terwujud, maka dapat menjadi langkah besar dalam rangka mewujudkan kedaulatan pangan Indonesia. Semoga tayangan kali ini bermanfaat. Jangan lupa subscribe, komen, dan share.
Halo.
Tes. Halo.
Indonesia is one of the most biodiverse countries in the world, which has a wide variety of ecosystems, species, and its genetic uniqueness. Treasures that we need to protect, preserve, and nurture with the help of science and technology. And that journey begins here. A place where we can inspire each other to innovate with integrity. A place where we feel inspired by the journeys of our forefathers that opened up the world's horizons with such a strong will, learning together. A place where we must strive, because we know that what we fight for is not only for ourselves, but also for our parents, our family, and the society that we live in.
Mbak, saya mau kirim uang untuk orang tua saya di Gresik. Ini Mbak, terima kasih ya.
With a strong belief, whoever we are, wherever we came from, and regardless of our background stories, we must believe that we can do it. Even though we realize there is no road without obstacles, challenges will always be there as a part of the journey. but we have to rise again. Bracing our hearts to reach our goals. And learning to understand and respect the important values of a process that we have to go through with sincerity and honesty.
Do I check?
Empowering society in the whole country. Spreading inspirations to the world to innovate with integrity. Because we are IPB University. Inspiring innovation with integrity.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Our warmest welcome from Bogor, Indonesia. We are joined here today in the joint webinar CO-SUPERVISOR International 2023 Postdoctoral Program IPB University, hosted by IPB University postgraduate student and animal breeding and genetic student community.
Representing IPB University, I would like to personally welcome our Dean of Animal Science Faculty, Dr. Insinyur Idat Gali Permana, Vice Dean of Animal Science Faculty, Professor Irma Isnavia and Dr. Sri Suharti, the honorable expert speakers, and our beloved lecturers and participants.
Well, ladies and gentlemen, animal breeding is a crucial component of global food production. And with the increasing demand for high-quality animal products, there is a need for more efficient and precise breeding methods. In recent years, the use of molecular technology has emerged as a game-changing tool in animal breeding, providing more accurate and effective methods for genetic selection. To explore the latest developments in this field, we are excited to host the joint webinar CO-SUPERVISOR International 2023.
The aims of this webinar are to bring together leading academic scientists, researchers, and research scholars to exchange and share their experiences and research results on all aspects of animal breeding and genetics. The attendees will gain a comprehensive understanding of the potential of molecular technology in animal breeding and its impact on the future of agriculture and food production.
All right, ladies and gentlemen, before we start our session today, allow us to introduce myself. My name is Nadia and my peers.
My name is Dafa.
And today we are entrusted with the role of the host in this webinar. We hope that during this webinar session, all the participants will be able to turn on the camera and stay muted on their microphone.
Okay, allow me to announce the webinar guidelines for today. First, use the virtual background provided by the committee. Rename your Zoom username according to the following formats: institution _ name. Mute the microphone, especially during the presentation of speakers. Questions from all participants are delivered using the raise hand, Q&A, or chat feature in Zoom. Attendees are not allowed to write opinion or information that is offensive, racist, or immoral in nature. Ladies and gentlemen, our agenda today consists of welcoming speech from the Dean of Animal Science Faculty, and then we continue to photo session. Then we will begin our first webinar session about molecular marker on livestock improvements, followed by the second session about molecular tools on animal breeding. Lastly, we will close with door prize winner announcement.
Well, ladies and gentlemen, let's start our webinar with praying that will be led by Mr. Amin. To Mr. Amin, the spot is yours.
Assalamualaikum warahmatullahi wabarakatuh. On this occasion, hopefully the event will run smoothly. I will lead the prayer according to Islamic law, and all of you pray according to your respective belief. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Bismillahi ma syaa Allah la yasukul khaira illallah. Bismillahi ma syaa Allah la yasrifus su'a illallah. Bismillahi ma syaa Allah ma kana min ni'matin illa fa min Allah. Bismillahi ma syaa Allah la hawla wa la quwata illa billahil aliyyil azim. Allahumma salli salatan kamilatan wa sallim salaman taman ala sayyidina Muhammadin litanhalum bihil unqad wa tanfarju bihil qurab wa tuqda bil hawaij wa tunalu bihir raghaib wa husnul khawatimi wa istasqal ghamamu biwajhil karim wa ala kulli mahdi wa nafasim bi adadi kulli ma'lum lak. Allahumma salli wa sallim wa barik ala sayyidina Muhammad salatan tunjina biha min jami'il ahwali wal afat wa taqdi lana biha jami'al hajat wa tudahhiruna biha min jami'is sayyi'at wa tarfa'una biha indaka lad darajat wa tuballighuna biha aqsal ghayat min jami'il khairati fil hayati wa ba'dal mamat. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzaban nar. Wa sallallahu ala sayyidina Muhammadin nabiy ummi wa ala alihi washabbihi wa barik wa sallam. Subhana rabbika rabbil izzati amma yasifun wa salamun alal mursalin walhamdulillahi rabbil alamin.
All right, thank you, Mr. Amin, for leading the praying.
We move to our next agenda. It is customary in Indonesia to start an event with our national anthem. So we will first play a video of the Indonesian national anthem, followed by the hymn of IPB University. Please remain muted throughout the video. To the committee on duty, the time is yours.
Now we will begin our next agenda, the welcoming speech from the Dean of Animal Science, IPB University, Your Honorable Dr. Idat Gali Permana. The time is yours.
Thank you, MC. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, distinguished speakers, Professor Goran Andersson from University of Agriculture Science, Sweden, Dr. Muhammad Jasim Udin from Murdoch University, Australia, and also Mrs. Ratna Solatiha Harahap and Mrs. Dwi Lestari, Dr. Candidate from IPB University, distinguished lecturer, there are Professor Roni Rahmanur, Professor Asep Gunawan from Division of Animal Genetic and Breeding, Faculty of Animal Science, IPB University, all of participants, ladies and gentlemen. On behalf of the Faculty of Animal Science, IPB University, I am pleased to welcome you to the joint webinar for Supervisor International 2023 Postdoctoral Program IPB University. With the interesting theme, I think, the application of molecular technology in animal production and disease resistance. This event is organized by postgraduate student and animal breeding and genetic student community IPB University. As we know that the application of molecular technology has revolutionized the field of animal science and has transformed our understanding of animal genetic, genomic, and biotechnology. This advancement have opened up new possibilities for animal production, disease resistance, and also for food security. The potential benefits of this technology are extensive, and their applications are numerous, ranging from developing new vaccine to improvement of animal breeding program. This webinar brings together scientists, researchers, and students from diverse fields and backgrounds, all with the common interest in applying molecular technology to animal production and disease resistance. This event provides an opportunity for us to learn from each other, to share the result of our research, and to discuss the latest development of the field. So the objective of this event is to deepen our understanding of potential benefit of molecular technology in animal science and to identify the challenge and opportunity they present. I encourage everyone to actively participate in the discussion, ask questions, and share your insight. Finally, I would like to express my sincere gratitude to Directorate General of Higher Education, Ministry of Education, Culture, Research, and Technology for generous postdoctoral grant. I would like to extend also my thanks to the International Cooperation Office, IPB University, and the Animal Breeding and Genetic Student Community, IPB, for their effort and dedication in organizing this event. I welcome all of you and wish you a fruitful and enjoyable discussion. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Idat, for the welcoming speech.
Before we continue to our next agenda, we will have a photo session. To the on-duty committee, I'll let you to comment this photo session.
All right, to all participants, please open your camera. Okay, in count to 3 from slide number 1. 1, 2, and 3. Slide number 2. 1, 2, and 3. And slide number 3. 1, 2, and 3. slide number 4. 1, 2, 3. And slide number 5. 1, 2, 3. And last slide. 1, 2, and 3. Right. Thank you.
Right. Now we will continue to our main agenda. But before we start, let me introduce our remarkable moderator that will lead the first session of this webinar. So, our first moderator will be moderated by Dr. Ihsan Suhendro. He has a lot of experiences such as farm stocking, slaughterhouse, and akikah of Saung Domba Bogor in 2017. And he had been supervisor breeding division of Java Purwakarta in 2018. He also a lecturer in University Tulang Bawang Lampung from 2033 until now. He also a postdoctoral at PB University in 2023 and postdoctoral brain zoology Terapan Bogor in 2023. Without take any longer time, so let us welcome our remarkable moderator. Hello, Dr. Ihsan, are you there with us?
Hello, I'm here.
Okay, so for the first webinar, we'll lead by Dr. Ihsan. So yeah, for now, this time and screen is yours.
Okay, thank you for the chance. Hello, everyone, ladies and gentlemen, distinguished guests, and esteemed participants. First of all, thanks God of the Almighty, because of His blessing and His mercy, we all can gather here on this sunny afternoon. In this chance, I am Ihsan Suhendro. I am from Lampung and I as postdoctoral researcher on IPB University. Assalamualaikum warahmatullahi wabarakatuh. Now, for the first session, there will be two speakers who will so much be telling you about the molecular marker on livestock improvement. In recent years, we have witnessed remarkable development in the molecular technique that have revolutionized the way we approach the animal production and disease management also. Through the application of molecular technique, we can now unlock the secrets of the animal genome, like identifying the gene association with the certainly desirable traits. Therefore, these tools have provided us to improve the breeding program, animal health, and sustainable production system. Today, we have two speakers from IPB University as the candidate of doctors who will present their research findings, success stories, and the future prospect related to the application of molecular technique in animal production and disease resistance. Their expertise will tell us how to make this molecular technique to improve the animal production and disease resistance. The first one is Ratna Solatia Harhap, doctor candidate, who will share his latest research on function of cytochrome family gene on lamb quality. And the second one is Dr. Candidate Dwi Lestari, who will share his latest research with the topic of function of BLB2 gene on chicken disease resistance. Lastly, I urge you all to seize this opportunity to expand your knowledge for the new connection and be inspired by the cutting-edge advancement presented today. In addition to the enlightening presentation, we encourage active participation from all of you through the interactive discussion and knowledge sharing. Okay, here we go. First, the first speaker, Ratna Solatia Harhap, time is yours.
Okay, thank you, Mr. Ihsan, for guiding the introduction. Good afternoon, everyone. First of all, I would like to thank you to the webinar committee for inviting me as a speaker in joint webinar CO-SUPERVISOR International. So what I'm going to speak today about the summary of functional of CYP2A1 gene on LEM quality. Okay. Okay, this is the introduction. Meat is one of popular animal husbandry product and eaten as source of protein. Meat is valuable source of high biological value protein, iron, vitamin, and mineral. White and meat, both animal-based protein that have their own unique taste and health benefit. Red meat, as any animal protein obtained from beef, pork, and sheep, and white meat comes from poultry and that is lighter in color than other parts when cooked. With Indonesia being a Muslim majority country, halal food influenced the buying decision of many Indonesian consumers. Lamb could used as meat sought after beef to support food consumption diversification. This is the lamb market trend. The total lamb production has increased in the last 5 years by 16,56,000 tons. This amount is still not enough to meet the demand for lamb meat consumption, which also record an increase of 7,74,000 tons. These are some meat producing sheep breeds in Indonesia. The sheep are part of local sheep group as Jonggol sheep, Javanese tin-tail sheep, Javanese fat-tail sheep, and Garut sheep. And then there are crossing breed sheep, including Garut composite, Barbados cross, and Agrina composite. The contribution of lamb meat to meeting the protein needs of livestock in the smallest, namely 4%, compared to the other livestock such as pig, 10%, and chicken 65%, and beef 21%. Why the reason for the low consumption of meat in Indonesia? Due to the assumption is circulating in society that is lamb high of content of saturated fatty acid, and the lamb has distinctive smell of lamb, and the price of lamb is pricey. An effort to increase the production and quality of lamb meat are direct solution in solving this problem, improving lamb meat quality, including carcass weight, physical quality, and chemical quality. This is are important to increase consumer taste for lamb meat. Nowadays, mostly the genetic quality of sheep is improved by classical breeding methods and support by various molecular to increase the rates of genetic gain for production traits of interest. Indeed, meat quality rates could be improved by using useful molecular markers with significant and positive effect on these attributes in marker-assisted selection. Meat quality is quantitative trait that's controlled by many pairs of gene or polygenic, and the heritability of lamb meat quality range 0.15 to 0.30. This indicates selection will be more effective to improve genetic merit for meat quality traits. One of the genes we have studied in the CYP2A1 gene or cytochrome, the CYP2A1 gene is located on 22 chromosomes and consists of 9 exons. Single nucleotide polymorphism of CYP2A1 gene had a significantly association with meat quality, its pH value, tenderness, as well as fatty acid composition. The CYP2A1 gene has been reported for several properties related to androstenone and scato level in pig, flavor odor in sheep, and fatty acid and lamb quality in sheep. As a member of the oxidative cytochrome family, this YP2A1 gene oxidizes a variety of small molecular endogenous substrates such as cholesterol biosynthesis, ethanol formation, as well as biotransformation of exogenous and xenobiotic. So this is how to know the effect of CYP2A1 on lamb quality. First, we can do a polymorphism study. This study to determine the diversity of genes, whether mutation occur. And the second is association study. This study to analyze the relationship of a gene with a trait to be observed. And the third is gene expression. This is to analyze the expression of a gene on a trait to be observed. First, identify CYP2A1 gene polymorphism study. We can doing DNA extraction and PCR amplification, PCR RFLP, the electrophoresis and UV transilluminator. The second, association study of CYP2A1 gene with lamb quality. The result of genotype polymorphism are related to the phenotypic we observe. In this case is carcass characteristic and fatty acid composition. Analysis data we can do with Minitab or SAS software. That is the CYP2A1 gene expression study, including selection of mRNA based on genotype and phenotype, RNA extraction and reverse transcriptase PCR, and cDNA quantification using KRT-PCR. So this is the result of genotyping polymorphism. We can see the CYP2A1 gene had polymorphism in Indonesian sheep, including GG, GT, or TT genotype. And then the CYP2A1 gene polymorphism with 3 genotypes found in crossbreed sheep and local sheep. Our population composition of allele and genotype is quite balanced. However, the GT genotype in our population is still dominant to be compared to GG or TT genotype. The CYP2A1 gene had a polymorphism and significantly association with carcass characteristic, including leg part, total muscle, and intramuscular fat. This is, we can see, the CYP2A1 gene polymorphism had significantly association with parameter of fatty acid, including saturated fatty acid, unsaturated fatty acid. We can see the crossbreed sheep had a fatty acid total higher than the local sheep, but for polyunsaturated fatty acid, the crossbreed sheep had a higher than local sheep. Third, the expression of CYP2A1 gene. The CYP2A1 gene was differently regulated among the sheep with higher effect of GG genotype. Quantitative real-time PCR analysis showed the abundance of CYP2A1 gene transcript in animal with divergent lamb quality in the liver. The CYP2A1 gene has an upregulate mechanism on lamb meat carcass characteristic and physical properties. The CYP2A1 gene has been widely reported for regulating skeletal intestinal in porcine liver tissue. The role of CYP2A1 gene in oxidation process is an essential basis that this gene can affect meat quality. So the implementation of the result of this study can be done by the selection and crossing. Selection using genetic marker of CYP2A1 gene with TT genotype can improve the quality of lamb meat. Sheep with the TT genotype can be produced by crossing homozygous TT genotype with homozygous TT genotype. Breeding sheep with homogeneous genotype for its gene is important so that sheep cross to produce the designed genotype can continue. Difference in the parents in the crossing process can also determine the phenotype of the result offspring. This due to the influence of sex on certain genes. Meat quality is a trait is generally inherited by family parents who inherit genetic material from cell nucleus or nucleus DNA and mitochondrial or mitochondrial DNA. So maybe that's all from me. I'm back to moderator. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Mrs. Ratna Solatia, for your information and your presentation. And I'm delightful to invite the second speaker of Dwi Lestari, who will be presenting the ideas of disease resistance. But before that, I will to inform you about the curriculum vitae of the first moderator. Excuse me, the first slide. Previous, okay. Ratna Solatia Harhab is a master and PhD degree in animal science of IPB University. She is an awardee of PMDSU scholarship, and she have so much of publication, like 5 or 6 publication in Cinta, also Schemagood, quartile 1 and quartile 2, yeah. There is so much of publication. And the second speaker, okay, next slide, please. This is Dwi Lestari, doctor candidate at IPB University. She is also bachelor, master, and PhD degree on animal science of IPB University. She has awardee on PMDSU scholarship. She has achievement on best presenter in 3rd International Conference of Animal Science and Technology in Hasanuddin University. She has so much of publication in Scopus and also in Cinta Journal National and International. Okay, this is Dwi Lestari. Time is yours.
Okay, thank you, Dr. Ihsan. Let me share my screen. Okay. Okay, thank you. Good afternoon, everyone. It was an honor to be able to attend in this wonderful seminar. As mentioned before, my name is Dwi Lestari. I am a doctoral student at IPB University. So today I will share some part of my research. So I hope we can share our views on application of molecular technology in animal production and disease resistance. So my research topic is about the major MSc class 2 gene BLB2 in disease resistant trait of Indonesian local chicken. Disease in chicken is caused by the immune system cannot fight and eliminate pathogens that infect the body. So pathogen can come from an hygienic environment that causes virus or bacteria that become pathogens. A bad environment can cause several causes such as an increased morbidity and mortality, and also decrease the profit. Therefore, an evolution related to the disease-resistant trait in chicken needs to be carried out. There are variant chicken breed in Indonesia, which have the respective advantages. Some are faster growth, some are disease-resistant, but crossbreed is expected to have excellent trait of both. IPBD1 chickens are local Indonesian chicken from F1 crossbreed male of Pelung and Sentul chickens with F1 crossbreed female of Kampung chickens and broiler parent stock strain crop. Based on Ulupi et al. 2016, crossing with broiler chickens resulted in a decrease in disease-resistant trait in IPBD1. The effort to improve the quality of IPBD1 chickens continue to be made until now. One of the efforts to improve the quality of IPBD1 chicken is the formation of new candidate line, IPBD2 chicken. The formation of IPBD2 chicken was done by phenotypic and molecular selection. Phenotypic selection is based on several criteria such as IgG concentration and ND antibody titer concentration. The disease-resistant trait has a heritability value of 0.2 until 0.3, which is categorized as moderate heritability. It means that selection will be more effective for improving disease-resistant traits in IPBD2 chicken. The selection can be done by utilizing potential genes that control disease-resistant traits. The major histocompatibility complex, or MSC, region is known that highly polymorphic and is associated with immunity traits. The MSC play significant roles in host immune surveillance and is a great importance in disease resistance. The MSC region in chicken is located on chromosome 16, which is divided into 3 classes, class 1, class 2, and class 3, which is class 1 and class 2 MSC included the BF and BL genes, which determine resistance and susceptibility to various diseases. Based on Rocha and Nefgis 2008, MSC molecule have a role in the process of antibody formation together with CD4 molecules. MSC class 2 molecule present antigen to CD4 T helper cell and help control the B cell differentiation in production antibodies. Mapping of the encoding chicken gene suggests that the BLB, the gene for MSC class 2 beta chain, is positioned candidate gene for disease-resistant traits. BLB2 gene pertaining to immune defense is regarded a major gene and expressed in a large variety of tissue in relatively high quantity in chickens. So how can be the BLB2 gene can help the selection of IPBD2 chicken? The first one is by polymorphism analysis by Sanger sequencing to get marker information of SNP polymorphism of this gene. The second one is association study with IgG concentration and the antibody titer. And the last one is gene expression using qRT-PCR method based on difference in high antibody titer and low antibody titer. So here the result, the length of amplified fragment of BLB2 gene is 871 base pair. But in amplifying the BLB2 gene, there are some obstacles such as the amplification results showing double bands. As Lee et al. 2010 state that the primer designed to amplify the exon 2 of BLB2 gene couldn't amplify perfectly the exon. But in this research, the addition of 5% DMSO can change the double band into single band in the BLB gene amplification process. And based on the sequencing result, there are 13 SNPs of BLB2 gene in IPBD2 chicken. And some SNP in BLB2 gene are thought to cause change in amino acid. Furthermore, for BLB2 gene association, 4 out of 13 BLB2 gene SNP were significantly associated with IgY concentration, but none of the 13 SNP were significantly associated with ND antibody titer. And for the BLB gene expression, IPBD2 chickens with high antibody titer have higher expression when compared to the IPBD2 chicken with low ND antibody titer. Based on Goni et al. 2009, high delta CT represent low expression, while highly expressed gene have low delta CT. The result of this study can be applied in the selection process of IPBD2 chicken, such as the first one is using potential SNP that associated with IgY concentration in the selection of local chickens, especially IPBD2 chicken with high IgY concentration. The second one is crossing the chicken with AA genotype with each other to produce 100 chicken with AA genotype. That's all for my presentation. Thank you for your time. It is my honor to present my research. Thank you for the time, and I open to any suggestions and questions in this seminar. Thank you.
Okay, thank you, Ms. Ratna Swatya Harhab and Ms. Dwi Lestari. Okay, ladies and gentlemen, we have heard the material presentation from our valuable speaker, Dwi Lestari and Ratna Swatya Harhab. We get some encourage of information of genetic molecular, which is so much good. The molecular widespread to the all of aspect knowledge and biological topic. Yeah, like we can provide the information from the disease Also from animal production. Usually it can be also using in what is the doctoral of the criminal identification of the criminal. But in this topic, we will to invite the participant to ask the question on share your opinion about this topic agenda, and I invite you if there are some of question from you. Yeah, anybody, you can raise your hand. In this webinar, we have some outdoor prize, yeah, for your question. If any question and any suggestion or opinion for this topic, you can share with us. Okay, I will to ask the question for Ratna Saldia Harhab before, yeah. For the implementation of SHIP on the meat quality on lamb, approximately how much years we can get the proper of lamb quality to get lamb quality from the SHIP, which is we now have lamb quality so and we will to make proper for lamb quality. So how many years we want for that? Yeah, Ratna, the is yours.
Okay, thank you for the question. To get sheep with higher lamb quality, we can do a crossing by fifth time. First, we can cross the sheep with TT genotype of CYP2A1 gene with the TT genotype CYP2A1 gene. This crossing we can do for parental one, and the crossing will be more doing for fifth knowledge. Thank you.
Okay, thank you. The second question for Dwi Lestari, I want to give your question. Compared to animal production, why you choose the animal health, the disease resistance? Why the topic so much important for you rather than the animal production?
Okay, thank you for the question. The first one, because our local farmer, we know that they grow the chicken in unhealthy condition, unhealthy environment. So it can be lead the disease to the chicken. So it can be decrease the profit, it can be increase the mortality and morbidity also like that. So we want to make a new line chicken IPBD to chicken with a good disease resistance, better than the ancestor IPBD1 chicken. Thank you.
Okay, thank you. For all the participants, for your information, the worker of this topic is IPBD1 and lamb quality. We will make the breeding of chicken and lamb for the future of our animal production in IPB University. So in this topic, in this seminar, we get the participant from all of Indonesia, yeah, in West, in East also, yeah. I will to invite the speaker from Papua maybe. Apri Irianto, you can give your opinion from you. I invite Apri Irianto. Any question from you? Or any participant, you can give the question from you. Okay, there is a question in chat room, yeah. From the first Mrs. Margawati. She asks, are those genes also work for meat quality other ruminant animals? Yeah. The question for Ratna Soratia. Ratna Soratia?
No.
There's question from Margawati. Are those genes also work for meat quality of other ruminant animals, or only in sheep animals?
Okay, because the lamb quality is a polygenic trait, so the lamb quality is affected by there are gene, there are in KIF gene, DGAT1 gene, and CXCL12 gene. Maybe that's all.
Okay, Miss Margawati, have you
not sure.
any response?
This also work for the cattle, for example.
Yeah.
Others work also for the cattle, for example. Mbak Ratna.
Hello, Miss Margawati. Can you repeat your question?
Already. Are those genes also can be used for the cattle, for example? Because you use sheep, isn't it?
Oh, yes. This cytochrome-2A1 gene is reported to be effect in lamb quality. Besides that, this YP2A1 gene had affected to pigs and goat. So this gene can use to the farm gene other.
So I can use it for the cattle, for example.
Yes, you can. Yeah, you can get. You can use the CYP2A1 gene for the cattle.
Okay, thank you.
Okay, thank you for the question from Ibu Endang Tri Margawati. For the second question, I invite Miss Irma from Kementan. You can ask the question.
Hello.
Yeah.
Okay. Thank you. My question is about the disease resistant in some local breed chicken. How if we cross the local this local breed with the other breed? For example, the offspring are heterozygote and different with the parental. Is the trait of disease resistant
Okay, Dwi, you can answer the question.
Okay, thank you to Mrs. Irma. The first one is the trait disease resistant controlled by a single gene or a polygene? Of course, the disease resistant trait is controlled by polygene. My research is only little part of the polygene that's control the disease resistant. And the second question is, and there are the phenotypic difference in crossbreed between local parents who have resistant characteristic if the offspring are heterozygous. Yes, there are phenotypic difference, of course, especially in crossbreed chicken that never selected before. Thank you.
Okay. Mrs. Irma, any response from you?
So this trait is expressed in homozygote or heterozygote mechanism? Because we need to select the progeny in order to have the trait that we want to have this trait.
For the offspring is we cross the IPBD2 chicken between the generations. So I think it is heterozygous between the IPBD2 chicken.
Okay, thank you.
Okay, thank you for the question. The third one is from Universitas Musamus. I invite Desmina Huta Barat. Time is yours. Okay, I will say the question. From Desmina Huta Barat for Ratna Soratiarat, there are any different of fatty acid composition for different genotype of lamb on Miss Ratna research? Any differences of fatty acid composition or not? Please explain.
Okay, thank you for the question. Allow me to show my slide. This is Miss Jessima. You can see my slide. This is the association of CY2A1 gene with fatty acid composition. We can see there are fatty acid composition on crossbreed sheep and the local sheep. For the saturated fatty acid, we can see the crossbreed sheep and the local sheep with the same of saturated fatty acid. But for the polyunsaturated fatty acid, we can see the higher of polyunsaturated fatty acid in local sheep, but the fatty acid total, the crossbreed sheep had the higher of fatty acid total in the fatty acid. Maybe that's all, Miss Desima.
Okay, Miss Desmina, any response from you? Okay, thank you. For the information, for the fourth question from Tulus Maulana, ya. Mr. Tulus, you can show your face. Okay, you can ask the question. Mr. Tulus.
Okay, thank you, Mrs. Natna. I have 3 questions for you. In this, for CIP genes in your research, is this gene researched by RNA sequencing or by reference? And second question, there is any other gene candidate to have major function in meat quality or meat tenderness? And the last question, in your research work and then an RNA study, do you have plan to analyze in proteome approaches? Thank you very much.
Okay, thank you, Mr. Tulus, for the question. For the first one is CY2A1 gene. This gene, yes, this CY2A1 gene based on RNA sequencing research. So for the polymorphism study, we can get a SNP by RNA sequencing or sequencing Sanger sequencing. And the second, there is any other gene candidate have emerged? Yes. The LEM quality is a polygenic trait that is a gene Carwell or a gene Calipets. If the gene is showed in LEM quality, so the LEM quality have a higher value because the Carwell and Calipets gene is a major gene for the LEM quality traits. And the third one is yes, for the analysis of proteome approach, we can use RNA for the expression study and the DNA for the association study. But if we get to know the expression, so we can use the RNA sample for our tip. Maybe that's all, Mr. Tulus.
Yeah. Mr. Tulus, any response from you?
Enough. Enough. Thank you.
Okay. We have still 15 minutes to Q&A session. So we have the question from Valencia Angelin. Time is yours.
Okay, thank you so much, Mr. Ihsan. Okay, I'm Valencia Angelin from IPB University. I want to ask a question for Ms. Ratna Solatiha. Is that another types of CYP gene in your research, like CYP 1A1 or 1A19 or etc.? Because in my research, have CYP 19A1 and CYP 19AB. And in my research, it's a type of gene CYP, it's different function and organ location. So can you explain about it? Thank you.
Okay, thank you, Mrs. Valencia. In our study research, besides the CYP2A1 gene, we also study about the CYP2A1, CYP2A6 gene. The CYP2A1, this cytochrome, it's a many gene family. Besides the CYP2A1 gene, we can use the CYP2A6 gene in our study.
Okay, thank you.
Thank you.
Okay, thank you for the question. Yeah. So the research from Ratna is about claim quality from association of CYP, cytochrome gene, family genes, and Valencia also working in cytochrome family genes, but in catfish. Yeah. So this molecular technique have a widely spread to all of the organisms, invertebrata, all bacteria. So this is an interesting technique you can use. So there are any questions from the others? We still have 10 minutes for the question. Okay, thank you for all of your commitment, all of your participants in this topic. I will say thank you for the speakers, our valuable speakers, Ratna Solatiha Rahab and Dwi Lestari, for your work in research and presentation in here. So thank you for all our participants for your question to the speakers and for your opinion for our speakers. And this is the last of from me. Thank you. If there are some mistake, I fully please forgive me, and thank you for all of the participants. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
I give to MC.
All right, thank you, Dr. Ihsan, for leading our first webinar session. Before we move to our next agenda, let us give a certificate as a commemoration and grateful from us to Dr. Isan. So yeah, Dr. Isan, here's the certificate for you. Okay, pardon. So the first certificate will give to Dr. Candidate Ratna Solatiha Rahab. Congratulations for your accomplishment. And next, is there any certificates?
Maybe there's some mistake in Ratna name. Solatia or Solatiha Ratna. Solatia. SH. Okay. Okay, for the second certificate.
All right. For the second certificate, I would like to give to Dr. Candidate Dwi Lestari. So congratulations for your accomplishment. Next. Okay, that's all. Okay, so yeah, thank you once again for our remarkable moderator and our speakers. Okay, next, I also want to give a certificate to our moderator. So for the operator, please show the certificate. All right, thank you, Dr. Isan, for leading this first webinar session. So yeah, this is for you. Thank you.
Thank you.
Okay, ladies and gentlemen, before we move to our second webinar session, we will have an ice breaking. So in this ice breaking, we will play a game that will challenge your brain to think about it. And we have also a prize, a huge prize. So yeah, I hope that all participants will actively take a part of this ice breaking and will get opportunity to get a prize. So for this session, I will let my peers, Miss Ega. Hello, Miss Ega. Are you there with us?
Yes, I'm here.
Okay, so what's the games about?
Actually, we have two games here. The first one is guess the song, and the second one is guess the picture.
Oh, nice. Okay, so yeah, please show about the game to us.
Okay, good afternoon, everyone.
Hello, good afternoon.
Good afternoon. It's so nice to see you, and I hope you're still doing good and still very excited about this conference, about this webinar. To just relax your brain in this afternoon, I have two games for you. The first one is guess a song. Anyone here loves music or update the new music nowadays? I hope you do, yeah? Okay. Can I share my screen? Oh. All right. The first one is guess the song. Since this is an international webinar, so the songs will be an international songs, which actually I don't really get into music, but yeah, let's just try it. All right, wait. So what you need to do, I will play a music and then a little bit part of the music, and then you need to guess what song is it, and as well as the singer. You can raise your hand if you know what song is it. Okay, you got the rules? Yeah? Okay, let me try. Okay. Anyone? Not yet? You can use the feature on the Zoom, the raise hand feature. Let me take more. The club isn't the best place to find the lovers. All right. Is there Miss Winnie, is it? Yeah, what is it, Miss Winnie? You know the song?
Shape of You by Iran.
All right, you got it right. Okay. Maybe some of the committee could write Miss Winnie's name. And let me play it one more.
I'm going on during this time. I feel there's no one to save me.
Anyone? All right, Mr. Bondan. Yes, please.
I guess Someone You Love by Lewis Capaldi.
Right. Very right. Okay. I guess just let's just one more song before we move on to the guest picture. A moment. Actually, I don't know this song.
Tastes like strawberries on a summer evening, and it sounds just like a song.
All right. Mrs. Rina Puspita. Miss Rina Puspita.
Harry Styles, strawberries. Watermelon.
Yeah. Very nice.
I'm talking about the lyric actually.
Okay. Good. Thank you so much for joining. And the next part, we have guess a picture. So what you need to do, I have some picture here. The version, we have two versions of it, the zoom out version and the zoom in version. Okay, so you need to guess what is it, and you can tell us by raising your hand also, or you can chat it on our chat room on the Zoom. Okay, let me try it. This one. What do you think is it? All right, Mr. Bondan.
I think it's crocodile.
Crocodile, all right. How about Mr. Lutfi? What do you think is it?
Crocodile head.
Crocodile head, all right. Mr. Ridwan?
This is something different. Alligator.
What is alligator, all right. Anything else? Alligator, crocodile. How about Miss Rina? You have different opinion?
No, I said alligator, maybe.
Alligator? All right, let me check on the chat room. Crocodile, crocodile. Okay, so most of you say it's crocodile or alligator. Let me give you. What's the answer?
Ya ampun. Kacau.
What?
Snail.
Call it, yes, snail crocodile, maybe. Okay, let me try another one. Right.
Giraffe.
Mr. Lutfi, what do you think is it? Or Miss Valencia? Any
I
opinion?
think giraffe.
Giraffe? Okay.
Yeah.
I think it's
How about-
tiger. Tiger maybe.
Tiger. Okay, tiger, giraffe.
Wah, motor nggak panggil guruna, besok dikelepak mana.
Miss Ade or Mr.
Ade, I think it's a bear. A bear.
Okay, bear, giraffe, tiger. Any other? Let me check on our chat, bro. Snake, elephant, giraffe, panda. Okay, let me show you the answer. Turns out it's a cat. Why is it so cringy? I'm sorry. Okay. The next one. What is it? All right, Mr. Ridwan.
It's looks like Oreos.
Oreos. All right. How about the others? Miss Rina, maybe? Or Miss Valencia again? Yeah? Miss Rina?
Oreo.
Oreos. Okay. Don't you learn from the past? How about Mr. Lutfi? Try to think
Chicken
more creative.
leg.
Chicken leg.
Chicken leg.
All right. Good. Give up, all of you?
Give some clue, please.
You can see the clues from the color of the Oreos. What do you think have like?
Zebra.
Think so? Who was saying zebra?
Irma.
Okay, let's check it out. Good job. It's a zebra.
Good.
Yes. Okay, next one. Come on. What do you think is it? Birds, chick, fingers of chicken. All right. Another?
I think it's bird.
Okay, let's see. It's lemons.
Nice.
Okay, I have some more here. Okay. Something in the ocean. It's black and white.
Whale.
Whales. Okay.
Yes. Look like whale.
Panda.
Dolphin. Panda. Okay. Whales. How about Mr. Lutfi? What do you think is it?
Dolphin.
Dolphin. Okay. Orca penguins. Good.
Willy.
Oh yeah, Willy. Yeah, let me show you the answer.
That is catfish.
Yeah, catfish. Okay. But this. Oh, I'm sorry. Okay. Cat. Oh ya, ini udah tadi ya. Sorry. Alright. Cat. Okay. Another.
Princess.
Princess. Good. Come on, be more creative. Rabbit. It should be a cat, but I think it's a dog. Okay. Okay. 3, 2, 1. Let's see. It's a rooster. Okay. I think I have 2 more here. Okay. Come on. Cattle, good. Another. Yes, Mr. Lutfi. Horse.
Seahorse. Seahorse.
Seahorse. Okay.
Seahorse.
Seahorse, right.
Seahorse.
Okay, seahorse. Let's see. Is it seahorse or cattle or a turtle giraffe? Okay. The last one. It's swimming, I think. You can see the legs. Duck, okay. And it's yellow.
Fish.
Frog, okay. Salamander. Right. Another?
Donut.
Salamander. Donut. That's a good guess. Anyone else? Cat. Okay. Fish. Yes. Okay. Anyone? That's all? Okay, let's reveal the answer. Lion. It's related to livestock. The clue. That is related to livestock. But yeah. You want to guess another? Cattle? Okay, cattle. Anything else? Chicken, right? Good, okay. Sheep, rabbit. Pig, lamb. Okay. Good job. Who's answering pig? It's a peacock. Okay. Thank you so much. And I will give it back to our MC. Thank you and enjoy the rest of the webinar.
Who is the winner?
Right. All right. Thank you, Miss Ega, for leading the interesting game. Oh, this is such an interesting one. Like, if we have chicken with sound meow, it's like a cute thing. So yeah, maybe Dr. Candidates here will like thinking about how to combine the sequence to make a meow chicken or a big size of lizard or something. Yeah, it's quite interesting. Okay, so for the winner, it will be contacted by our committee. So yeah, congratulations for the winner. So Dava, what we will have for the next agenda?
Okay, now we are fresh again. I think we are ready to our next second webinar session. Same as the previous session, let me introduce our remarkable moderator who leads today's second webinar session. To operator, please share screen the resume of the moderator. All right, for our second webinar session, Profesor Dr. Ager Asep Gunawan, SPT MSc IPU, will lead as a moderator for the second webinar session. Next, he is now a senior lecturer at IPB University. He had accomplished his education in bachelor degree of animal science IPB University at 2003, master degree of animal breeding and genetics in University of Bonn at 2010, doctoral degree of animal breeding and genetics, University of Bonn at 2013. He also had many experiences such as chief editor, chairman of Commission A, and secretary of Commission D at IPB Professors Council. And also he also wins many awards such as 113th Indonesian Innovation 2021 and also Innovator of the Year for Information Technology Development by Metro TV. So now I will call Professor Asep Gunawan. Are you there?
Yeah. Hello.
Hello, Prof.
Java. Thank you.
Hello, Prof. Asep. How are you today?
Yeah, I'm fine. And you?
Yes, I'm fine too. Okay, so I think this session will be led by you, and I will give you the time and the screen.
Okay, thank you for introducing me. So today I would like to see, are there Dr. Jasim is already here? Hello, Dr. Jasim.
Hey, hello, Asep. How are you?
Hello.
Can you hear
Yeah,
me?
thank you. So, yeah, good afternoon, everyone in the session of 2 session in the afternoon. So it's my pleasure to welcome you all again to this joint webinar, Co-Supervisor International Postdoctoral Program IPB University. And I am honored to serve as your moderator for session 2 for today. So as we gather here today, I believe that we all share a common goal, which is to engage in a meaningful discussion, sharing the idea and learn from one to another. The topic we will be discussing is one that of a great importance all to us. And I am excited to be a part of this conversation. So as your moderator, my role is to ensure that the event runs smoothly and that everyone has the opportunity to express their opinion and thought in respectful and constructive manner. So I will be keeping track on time, managing the flow and discussion, and facilitating the Q&A, question and answer session in the end. I encourage everyone to participate actively in this discussion, ask questions, and share your perspectives. Let us embrace diversity and different viewpoints and engage in meaningful dialogue that will help us gain a deeper understanding the topic. Before we start, I would like to remind everyone to respect each other, opinion, refrain, and from making personal attack engage disruptive and behavior. So I hope we already have, I hope that this event will be informative, insightful, engaging, and I look forward to hearing the idea that you bring here. And let me start by introducing our speaker here. We have 2 speakers in the section 2 for today who are renowned and expert in the field, in their field. With many years experience and well knowledge, and they are share with continuous individual and organization. They have been instrumental advancing and outstanding, and the related topic, and contribution a significant impact to our institution. The first speaker is Dr. Muhammad Jasimuddin. So he's already been here with us. He's a senior lecturer at Murdoch University, Perth, yeah, Australia. Is maybe our neighbor, yeah, and maybe now it's more afternoon, yeah, different time.
Yes.
We also have in the letter, and the second is Professor Goran Andersson. Yeah, is from the Swedish University Agricultural Science. So this speaker, we hope we can get impressive, yeah, knowledge, yeah, and also for us especially, yeah, we can get more information, yeah, related with the current understanding of the animal breeding and genetics. So before, without further ado, I know that you all eager to hear, yeah, their insight. Let me start first to introduce, yeah, the first speaker, Dr. Muhammad Jasimuddin. Yeah, allow me to brief introduce him, yeah, by the curriculum vitae, yeah. Muhammad Jasimuddin, PhD, he's a graduate doctor veterinary medicine in 2004 from Bangladesh Agricultural University. And then 2006, he graduated a Master Science in Veterinary Science, Bangladesh Agricultural University. And then in 2011, yeah, he got a PhD in Agriculture Science University of Bonn, Germany. So he had a lot of experience, yeah, and we know he has very productive scientists, yeah, which age index, yeah, 25, yeah. So we have it's very proud, yeah, to attend here. And then related with the experience in 2021, now, yeah, he's a senior lecturer, yeah, in veterinary virology of Murdoch University, Australia. Before, of course, he get a postdoctoral, yeah, in Germany, and also in Australia in University of Queensland. And regarding with dedicate his work, he got a lot award, yeah. In 2006, University Gold Medal, yeah, for distinguished in MS in veterinary medicine. And then in 2019, yeah, he got the Global Research Impact Award, yeah, in the third position at the university and in the first, yeah, faculty level of Bangladesh Agriculture University. Okay, that's from my point, yeah. For the next, yeah, the time is yours, yeah, to Dr. Muhammad Jasimuddin. Please.
Okay, thank you very much, Asif. Is it clear? Can you hear me?
Yes.
Okay.
It's very clear.
Should I get the control and share my screen, or do you want to control it? How should we start?
If you more convenient by share your own, maybe, yes. Yeah. Yes. The computer can get access, yeah.
Can you see my screen now?
Yes.
Can you see the presentation?
Yes, yeah, it's clear, yeah.
So can you see the full screen, or do you see half screen?
Okay. Can you maybe, this is, so it's not
so
full.
You want me to swap, right? Okay, let me try. How can I swap it?
Yeah, that.
And then that's the problem I may face. Okay. Can you see now? Is it okay now?
Yeah, we can see, but not in full screen. Yeah.
In full screen. Okay. So how can we see? Let me try. I don't know why it's showing the next slide. So that's probably a problem. Oh, sorry, because I'm not good at, How about now? Is it okay now? Can you see the full screen?
How are others? Maybe from the computer, is not full screen now?
Now is it full screen?
Okay. Not yet, yeah? Okay, if not, maybe can somebody help to...
I mean, is it now full screen? Can you see?
Not yet. So
Not
maybe
yet.
I try to help you.
Yeah.
If maybe it could be
Can you see now? Is it now full screen?
Yeah, full screen now.
Okay, that's good.
Yeah.
So, should I start, Asif, now?
Yeah, it's already. Yeah. Okay.
Thank
Please,
you very
the time
much.
is yours.
I'm extremely sorry for this little IT stuff. This is always happening with me. I don't know why, but it's always happening with me. So first of all, thank you everyone for joining today's webinar. And we are going to discuss today about the RNA sequencing in animal breeding. Let's be clear that RNA sequencing is a modern technique that we use not only for animal breeding policy, we also use a lot for disease diagnosis, for virus sequencing, for other animals and crop sequencing. So it has a lot of implication from evolutionary biology to the diagnostic. However, we animal scientists are using this technology for animal breeding. So it's my pleasure to talk about the RNA sequencing in animal breeding, but I'm not going to discuss in details about the RNA sequencing and how to include that in the breeding policy using junk data as well as very hard statistics or biostatistician knowledge. I will try to keep it very simple so that we all can enjoy it. So welcome to my presentation. And my name is Joshimuddin and I'm working in Murdoch University. If you want to know details about my work, you can visit my profile. So first of all, I want to acknowledge the Wajjuk people and Noongar Nation. First of all, RNA sequencing has a different name. Like some people call it RNA-seq, some people call it deep sequencing, some people will call it high throughput sequencing or HTS. However, some people will also call it NGS or next generation sequencing. I don't mind to call it RNA sequencing or high throughput sequencing, but I think NGS or next generation sequencing is a kind of misnomer because it is not anymore next generation. It is here and we are using it. If I want to tell you briefly, there are several platforms in the market. You will see different labs are using different platforms like MySeq, HiSeq, NextSeq, NovaSeq, or Solexa from Illumina. There is also sequencing platform from PacBio. You will also find very small, like a mobile size of a mobile from Minion, from Oxford Nanopore. You will also find Ion Torrent use a lot, which is marketed by Life Technology. You have also heard about Roche 454. So there are different platforms in the market and they have different chemistry. So they are not working in the same way. However, I'll get back to them again. So first, let's talk a little bit about the sequencing and breeding. So you want to put sequencing and breeding together. How is this working? As I told you, sequencing technology has been evolved so much, and we, the animal breeder, are getting the benefit of sequencing. Sequencing has a lot of different uses. If I want to talk about the use of sequencing, then I have to talk 1 hour or more than that. But it has a lot of implication, and we animal scientists are also using it for our purposes, for animal breeding. Why do we use it? We will discuss in details later. But first, let's talk a little bit about the phenotype. We are always aiming for the better phenotype, for the better production, better health, as well as like more milk production, more egg production. So we are always pushing the animal to produce more milk, more meat. I mean, we want to get the desired phenotype more and more. But you know very well that phenotype is controlled by genotype, the genetic makeup of the animal, as well as the environment and the interaction of all. So we call epigenome. However, we scientists are focusing on the genome, because if we can get the desired phenotype by twisting or manipulating the genome, that will be more interesting, because genetic changes are more permanent. That's why we push the genetics and we use the genetics to get the better phenotype. And how do we do that? When I talk about the genome, I actually talk about the genes as well as the polymorphism, like single nucleotide polymorphism, or what we call SNP. So if we know which genes and which SNPs are contributing or controlling the positive phenotype or desired phenotype, then we can do the association study. We can find out which genetic marker is positively contributing, and then those genetic marker or polymorphism or the genes we can use for marker assisted selection breeding. And this is how we can get the desired phenotype. However, as I told you that when I talk about the genotypes, genotypes means which genes are contributing, which genes are controlling the desired phenotype, which polymorphism or which genetic marker or single nucleotide polymorphisms are controlling the phenotype. We want to find out those. So when I talk about the transcriptome, we also mean the differentially expressed gene. We mean the splice variation, microRNA, long non-coding RNA, methylation, lot of things. So for the selection breeding, we need to know the transcriptome, the genetic makeup, as well as the polymorphism. And that's why we need the sequencing technology, because sequencing technology can generate huge data about the transcriptome as well as for the polymorphism. We will learn a little bit more later. That's why, as I told you, sequencing technologies has a lot of different uses, but we animal scientists can also use those to find out the genetic markers so that we can get animal with better phenotype. So if I want to tell a little bit about the history or background of the RNA analysis, because RNA analysis or RNA expression or detection is actually very important. So this is the basis of all our work. In 1950s or 60s, people used to do RNA blot. It's a kind of Western blot. We use Western blot for protein expression, and they use RNA blot or Northern blot for RNA detection. This is a very crude method because you need a lot of genomic material, as well as you have to run it and then electrophoresis, transfer it to the membrane, and then using the X-ray, you can see the band. But this is not giving a very meaningful data. So later on, we get the PCR. PCR is a fantastic technology. It is been using in science for research, diagnosis since a long time. Still, the PCR is, I would say, one of the most common technique we use in biotechnology or in medicine or in science overall. However, PCR has some limitation. It can handle, or you can use the PCR to quantify only few genes because it's time consuming. It quantifies gene one by one, or if you do multiplex PCR, maybe 10 genes at a time. But we want more because we want to know more about the whole genome of the animal. Then we got the microarray. Microarray is a kind of better version, and it can do it can quantify a lot of genes at a time. So we get the microarray. When I was PhD student, microarray was something, oh, this is the technology we all want, because this can just, quantify the whole transcriptome. However, microarray has also some limitations I will discuss later. So we get the next generation sequencing, or NGS, or RNA-seq, or deep sequencing, or high throughput sequencing, whatever you call. So I will call it RNA-seq. So we get the RNA sequencing to overcome all the limitation we had for microRNA. But still RNA-seq has some limitation. I will explain later. As I told you that we use a lot of QRT-PCR, quantitative real-time PCR, to diagnose or to understand the gene expression differences. You can see we have published a lot of paper and we use QRT-PCR. QRT-PCR you can use to quantify the expression of the gene, and you can compare between the good phenotype and bad phenotype. But PCR has some limitation. You can see you can do the expression study of few genes or a single gene at a time, and it has some other limitation. As I told you, it is not high throughput because it cannot quantify a lot of genes at a time, as well as for PCR you need the primer to bind with and to do the multi-replication. So for PCR you need the known genome. It cannot quantify the unknown genome or novel genome, as well as it is time consuming because you can quantify only one or few genes at a time. So the new technology come up in the market that is called microarray. As I told you, when I was PhD student, it was a sensation, microarray for genomic study. How the microarray work? The microarray, you see, this is a chip, and then in this chip there are a lot of small hole, and in there are a lot of probe, and these probe are actually short sequence. When you are adding your sample to this probe, and if this probe match up with the sequence in your sample, then they will bind and you can see the fluorescence. So you can isolate your genomic material, you can do the labeling and then hybridize with the probe, and then you can do the scanning, microarray scanner. And after some bioinformatics, you will get the expression, but it has also problem. Like it can quantify thousands of genes, that's okay, but it has low dynamics. Like it can only do the expression study, or if you want to do the array to do the polymorphism study, you have to use the separate scanner as well as the probe. So it has a low dynamics, as well as it cannot detect the novel transcript or unannotated transcript. Also, for the data analysis, you need the reference genome, the genome that we already know. So it has also some limitations. However, saying that, we still use microarray. I think nowadays the use of microarray is getting lower, but still some lab use microarray, and during our time we use a lot of microarray, and you see we use the microarray to understand the transcriptomic signature of host pathogen interaction. We have done a lot of work with porcine reproductive and respiratory syndrome virus, and how they interact with the cell, like blood mononuclear cell, as well as how this signature is different between the different pig breed. Like, as I told you, for all this work, we use Affymetrix gene chip, and this gene chip has like almost 400,000 probe, and this 400,000 probe together can represent almost 19,000 gene. So compared to PCR, it is a huge step to quantify thousands of genes at a time. But as I told you, microarray has some limitation because it cannot quantify the novel transcript, the unknown transcript. It needs the reference genome. So I now want to talk a little bit about the genetic marker. We have talked about the gene expression. Now we'll talk about the genetic marker or polymorphism like SNPs. So how did we work with SNP? Initially, we have done a very time-consuming job, like you sequence, you get your genomic material, then do the PCR, and then ligate that in the vector, grow the vector, then isolate your PCR product, and then you do the short sequence. And then you match up this short sequence with the known genome, and then you will find the genetic marker or the nucleotide polymorphism, because when you will blast or you will do the alignment of different sequence, you will see where there is some changes. I mean the polymorphism, like SNPs that we call the genetic marker. However, this is a very time-consuming and laborious job. And if you want to cover a whole gene, then you need several primer pairs to quantify and expression or sequencing of the whole gene. So it's also very useful technique, but it has limitation. Now, after the PCR product you get, you have to do it, the enzymatic restriction enzyme digestion. After that, you can run it in the gel and you can see the band. So this is how, in traditional way, we identify the SNP as well as we did the genotyping. As I told you that we used to do it, still we are using it. If you want to work with one or two polymorphism, one or two gene, this is still very economic, but it is a laborious job. And you can see we have published quite a lot of paper following this method. Like we have identified some very good candidate marker, genetic marker or SNP in IFI6 gene, as well as in Haysen F1A. You have seen in CD9, and lot of, I can even give you a lot of more example. By the way, I want to tell you that all the examples I will give you here, most of the example is from our group. We have done this work by ourselves. So you will see that me, Asif, we together have done a lot of job. So I will try to give more and more example using our own data. However, let's get back to the topic again. So we did the genetic marker identification, and then we did the genotyping using RFLP, restriction fragment length polymorphism. I told you this is a laborious job. It needs time, but still it's work if you want to handle with few genes and marker. But it has also limitation, as I told you. For this, it is time consuming, it is not high throughput, and it cannot be used for the novel SNP. So we can only do the genotyping if the enzyme is available and if the SNP is known. So we come up SNP chip array-based genotyping. As I told you, that PCR is working, but we come up with microarray that is working better. Here also RFLP is also working, but then we find out array-based genotyping. Because if you see, this is the array, it's a kind of microarray. And in this array, there are thousands of SNP or polymorphisms there, like in this Illumina porcine SNP 60 bit chip, there are almost 80,000 different polymorphism here. So when you will hybridize and you will do the scanning of your sample, if your sample have this polymorphism, you can identify. And when you will have a lot of data, then you can do genome-wide association study. We used to do single gene association study, single or few SNP association study. Now we have the SNP chip array-based genotyping. So we can do the whole genome-wide association study. So this is also a big step. However, it has also limitation. Like microarray, this has also limitation. This array-based or chip-based genotyping can only detect the SNP that is known. It cannot handle the novel SNPs or unknown SNPs. So if you have money, then you can do a perfect genome-wide association study like we have used in this paper. You can see, you can use the Illumina SNP chip to identify thousands of SNP, as well as you can do the expression study using Affymetrix array. So using this technique, you can do a very good genome-wide association study as well as whole genome expression study. But these are based on microarray for transcriptome study and SNP chip array for genotyping. So they both are lacking of novel transcript. As I told you, microarray cannot identify the novel transcript, as well as SNP chip array cannot identify the novel SNPs. So they are very good. They produce huge data compared to PCR and RFLP, but still they cannot identify the novel SNP. To analyze them, you need the reference sequence. So we want something better than them because science is evolving. So what did we get? After this, we get the deep sequencing or RNA-seq. This is kind of all-in-one that we want because it has broad dynamics. You don't need a separate machine to do SNP chip array and separate machine for microarray. The single machine or the single platform can do both the transcriptome study as well as to identify the SNPs. So it is kind of all in one, as well as this technique can also identify the novel, I mean, novel means novel. So genome as well as transcriptome as well as SNP. And it can also detect the unannotated gene. It can also detect the alternative splicing. It can also detect the novel SNP. And using this method, we can also do the genotyping. So we can do the sequencing for the gene expression, as well as we can do genotyping, like it called genotyping by sequencing. And you can get huge number of data, huge number of RNA transcriptome, as well as polymorphism. So it's kind of like everything you want from microRNA, as well as for SNP chip array, even overcoming all those limitations. It is very high sensitive, it is accurate, it can produce very high resolution map of the genome, as well as you need only low amount of starting materials to work with this, because sometimes you need to work with single cell. So this is very, very sensitive and it has a lot of use. So what we see that suddenly the microarray use dropped and the sequencing technology getting more and more popular. So we have actually catch up with the sequencing at this stage, like around 2008 and 9. Still by that time, we were using a lot of microarray, and we started to use the deep sequencing. So you see the microarray use is dropped, and the sequencing technique is getting more and more popular. And this data show up to 2016. If you can have the data till today, you will see microRNA use is almost getting to zero, and sequencing data is getting high, skyrocketing. Now, we will talk a little bit about the RNA sequencing. Up until now, I have discussed with you what is different technique and how we came up with RNA sequencing. Now let's focus on RNA sequencing. So RNA sequencing has also went through different developmental processes, because science is always evolving, always better and better platform are coming. So you can see different company has different platform, their chemistry is different. Like some platform will do the short sequence read, and some can read the long sequence. The machine is getting smaller and smaller and more powerful. So I'm not going to talk in details about the chemistry, but let's see how the RNA sequencing work. Remember, it has a lot of different steps. I have done it. If you have any experience, you know that RNA sequencing has a lot of different steps. And all these steps depend on the kit you are using as well as on the platform. Every platform have their different way of running, as well as they suggest to use different kit. So it has a lot of steps, but those steps depend on the kits and the platform. But grossly, you will have your genomic material. If you want to use the short sequence technique like MiSeq or HiSeq, then you need to do the fragmentation. And when you will do the fragmentation of the genome, you need to add the adapter and primed it. Then you have to do the cDNA synthesis, ligation with adapter for labeling, and then library preparation, library cleanup, library quantification, and quality check is very, very important. And finally, sequencing. It has a lot of different steps, and you need a good training. Since it has a lot of different steps, we see some other company are coming up with robotic to handle the fluid handling robotics, because when human will do a lot of steps, there is a high chance of doing mistake. So this fluid handling system can do it more accurately, and more precisely. However, all this fluid handling system is very, very expensive. Now, let's talk how do we get the fine data. If you do your NGS or deep sequencing, and next morning you will get millions of base pair A, T, C, Z. And what to do with that? It's not easy to handle millions of data. So you set up your deep sequencing, and next morning you come to office and you find huge data. Now you have to analyze it, you have to visualize it. And this is a very complex process. That's why we, the scientists, work with the bioinformatician, we work with the computer scientist, we work with the programmer. This is the beauty of science. We work together. The science is getting smarter and smarter. We are digging inside and inside, and we need more people from different expertise to work together. So analysis is very, I would say, complex, and it needs very good knowledge on the bioinformatics. And there are some analysis pipeline, like some analysis platform will be free, like Galaxy or R. Some will be, you have to buy, like Xenius. Of course, this will be free. You need to manipulate, and you need to know how to do the programming, because everything happening behind this pipeline, there are some program is running. So you need to understand those things. However, there are some analysis pipeline in the market, and believe me, it is getting smarter and smarter, easier and easier, more and more user friendly. So when we first used the deep sequencing in 2008 and 9, we need to hire a whole team. Like we appointed a PhD student who has no genetic background, but a pure computer scientist and programmer to work with mathematicians to burn all this data. But nowadays, you will see that people like me and like you with training can also handle this data. So it's getting easier and easier day by day. And when you will get all this data, you also need to submit this data. There are a lot of data bank, you can submit it so that you can use later, as well as your colleague can also use this data. This is very important to share your data with other scientists all over the world. This is how science grows. So, as I told you, it is getting more and more popular, as well as the cost is getting lower and lower. Believe me, when the sequencing technique first, I mean the RNA-seq first started in 2000, it was very, very expensive. When we first used the RNA-seq in 2008 and 9, we pay almost 30,000 euro to analyze or to sequence only 6 samples. And nowadays, 6 samples will be like almost $100 per sample. So the price has gone down, and that's why it get more and more popularity. The price has gone down, and as well as the handling of data is getting easier. That's why it is getting more and more popular. So in 2018 or 2008 or 7, when we did the first RNA-seq, we had no sequencer in our MySeq or HiSeq in our lab, because back at that time it was very expensive. So only the commercial setup we use. We sent the sample, they did the sequencing, and then they sent it to the computer scientist, and they analyze for us. Nowadays, you will find many labs, especially in the developed country, almost everywhere you will find the sequencer. So the sequencer platform is also getting cheaper and cheaper. Now, here you see we have used a lot of RNA-seq for our work. Like here you see for endostenon, as well as for scatol, Asep and me, we have used RNA-seq to understand the genomic signature, how the Bohr tent is controlled, as well as how the backfat is also controlling the Bohr tent. Later on, we also did more and more work with RNA-seq. Here you can see we have done the whole hepatic transcriptome analysis to understand the lamb tenderness or sheep meat tenderness, the fatty acid metabolism in sheep, also the fatty acid metabolism in chicken. We have also done RNA-seq to understand the liver or hepatic transcriptome, how they are affecting the mutton odor and flavor. So we have done a quite bit of work with RNA-seq, as well as we have used the RNA-seq to understand the porcine reproductive and respiratory syndrome virus and the dendritic cell interaction. I have shown you that we have used microarray to understand the PRRSB and the cell interaction. Later on, when the RNA-seq become available, we also started to use RNA-seq more than microarray to understand the host pathogen interaction, as well as to understand the genomics or genetic makeup controlling the phenotype. So I'll give you one of the example, because I'm not going to talk about all this 50 paper that we published with RNA-seq. One of the paper we can take an example, and I can show you how useful this technique is. Like this is the RNA-seq, and we published this paper. And then you can see it produces huge data set. So based on that, you can understand or you can identify the differentially expressed gene. If you can remember at the very beginning of my talk, I talked about that we need to understand which gene are differentially expressed, which polymorphism are controlling the phenotype. Then we can include them in selection breeding. So using this method, you can identify the differentially expressed gene, both known as well as novel. Then you can analyze all this data together to understand which pathways are involved to control this phenotype, because this is important to understand which pathway is involved, which genes involved, then which protein interactions are involved, because almost all phenotypes, only few exceptions, almost all phenotypes are controlled by several genes, several gene interaction. That means several protein and several protein interaction. So when you have all this expression data, you can actually calculate and visualize which genes are involved, which pathways are involved, which genes and protein and gene interactions are controlling the phenotype. Because you need to understand this. If you don't know which genes and proteins are important, how you will do the further work for selection breeding. And you see how powerful this machine, this platform can identify thousands, if not hundreds of SNPs. And then you can do the association analysis to find out specifically which polymorphism and which genes are actually controlling or contributing for the desired phenotype. You can see we have done more and more work. Once you have this sequencing data, you can use those data for further genotyping as well as for association study. We have worked a lot up until now. I have shown you all our paper. We have done huge work to understand which genes are controlling the phenotype, like meat quality phenotype, the disease resistant in pig, in sheep, but a lot of people are also working with other animals. More work has been done for cattle than the pig and lamb. Since my group and we work for pig and sheep, that's why I'm showing you the example from pig and sheep. Now, this is the one that's not our paper, but I can show you here that if you have the money, you can do your whole population's sequencing. You can use RNA-seq to sequence your whole population. Then you will find a high quality data to do both genomic association study as well as to identify all the SNPs. You can do sequence-based genotyping. So this depends on also the money you have for the project. Now, as I told you, we identify the genes as well as the polymorphism or genetic marker. Why? So that we can include them in selection breeding. But selection breeding is not a new thing. Like natural selection breeding is happening since hundreds of years. And then when we have this technology in our hand, we are also doing more selective breeding in industry scale so that we can get the desired trait or desired phenotype. As I told you, I have given you some example here with regards to the sheep as well as pig, but a lot of work has been done for cattle, for horse, a lot of work has been done for dog, for chicken to get better and better animals. When I say better, better means with more production. And this technique, the selection breeding, is also used a lot in plant and crop science, like for chickpea, for rice, for maize. They even use it more than the animal scientists use. Now, there are some downside. You know, when scientists are focusing so much about the production trait, like we want more milk, more meat, more egg. We are actually making the animal more prone to disease. The animal's body is compromising and animal is getting sick. They can get sick easily. That's why we need to focus on the breeding for resilience. I'll talk in a second. So there are some challenges. So RNA-seq is here and we are working fantastic, but there are still some challenges. What are those challenges? The availability. As I told you, that in developed country, in many labs, you will see the RNA-seq platform, but still in many countries, we don't see this platform a lot. But for science, we need to grow together, as well as the cost. Still, the initial setup cost is very high because all these platforms are very, very expensive. All these machines are very expensive. And later on, the running cost is also expensive because the preparation of all these kits and chemicals are very expensive. So if you don't run it regularly, it will not be economically viable. That's why the cost is still high. And I told you that we need more expertise. Still not many experts around who can do the analysis, who can do the whole RNA-seq. So it's actually all related. When the cost will go down, it will be more available. When it will be more available, we will see more and more expertise people will be involved. When there will be more expert people, the analysis will be easier. And another thing, we need all-in-one platform. As I told you, at the moment, there are several different platforms in the market. They all are doing fantastic job, but their chemistry is different. So if you want to do the shotgun analysis, you need like MySeq. If you want to do the long RNA sequencing, then you need nanopore. So we want even the better version, which can do in the same platform. So we need all in one platform. And the most important things, we need the global initiative, because nowadays, one group is working in Indonesia, another group is working in Australia, another group is working in Denmark, another is Sweden. But we all need to work together to find out the genes and the marker that is controlling the phenotype. If you want to get animal with better, resilient animal will be higher production as well as more robust to the disease. Then we need to work together, and that is now happening. You see the genome to phenome. So this consortium is burning and collaborating with scientists all over the world. And you see it is getting more and more fund, more and more people from all over the world are involving in this project. This is important. Like if you produce some data locally, and if it's not being used by some other scientist in the other part of the world, then this data has very low importance. So we need a global initiative to bring all the scientists who is using RNA-seq, because this is huge data. We produce the data, we publish paper, and we forget it. But that's not the purpose of science. We need to use all this data until we get what we want. Now, again, I will tell you, we are almost at the end. This is also our review paper we published in 2020, but still it is very applicable in today's scenario. Now we have the sequencer. We can identify which genes are important. We can identify which marker or polymorphism important, and we can incorporate that in the selection breeding. However, there are more fantastic techniques coming, like CRISPR-Cas9, genome editing technology. So if we know that this is the marker we need, this is the gene we need, this is the good gene, we can insert it, this is the bad gene, we can delete it using this CRISPR-Cas9 technology. So we need to merge all this technology together. And that is what's happening. You see, some brilliant scientists, they find out that NRAMP1 gene, which is contributing for the tuberculosis resistant, and they insert this gene and the cattle become tuberculosis resistant. And then you see CD163. This gene is the receptor for the porcine reproductive and respiratory virus, because all virus will bind with a specific receptor. So they find out that this virus is binding with CD163 gene. So you can now, using the CRISPR-Cas9, you can edit the genome, you can edit the genome to delete the CD163, and the pig will be resistant to porcine reproductive and respiratory syndrome virus. So this is how now we can work. We have all fantastic and smart technology to get to there. Now, I will just finish with this slide. So finally, we need both traditional systems, traditional technology, as well as the RNA technology to put them together, including the CRISPR-Cas9 or other smart technology which are coming, to put together to find smart, resilient cattle without compromising animal welfare. So we want animal with better production, better phenotype, without making them more sick or vulnerable to disease. That means we want also animal with resilience, as well as without any compromising of animal welfare. We should also respect the animal welfare beside the production, as well as disease resistant. Thank you very much.
Okay, yes, it's very interesting topic, and we get understanding very comprehensive, yeah, related with the topic, yeah, RNA sequencing, yeah, and then a lot of tool or technology that we can apply, yeah, in animal genetic and also breeding research. So we are very interest also, because from your explanation, yeah, it is on not only we get the explanation about the theory, but also you support also with the the wonderful of the research work. So I think it is very nice description, yeah, for us, and also so we can know what is the current situation, yeah, related with the update information about the animal breeding and genetic research topic, yeah, especially to the RNA sequencing technology. Okay, so the committee now we give the opportunity. Okay, so they have a discussion. Yeah, so they have a, of course, I think some of you, yeah, all the participants, maybe some have a question. So maybe this is a good opportunity, yeah, for you, yeah, to know, or maybe to ask, yeah, the question directly to Dr. Muhammad Jasimuddin. So maybe I give opportunity anybody to maybe deliver or maybe to ask directly question with the raised hand.
Yeah, I'm happy to take some questions. If you have questions, please you can
Yes.
ask me.
Okay. So, okay. So during maybe waiting for the direct question, so maybe I will read, yeah, from the chats, yeah. From Irma, yeah, from Ministry of Agriculture Institution. So I want to ask, yeah, whether any genetic marker application in marker-assisted selection has been applied commercially in the livestock industry. This is because the livestock selection system in Indonesia still maybe most, yeah, maybe not all, yeah, conventional, because such as based on specific phenotypic characteristic, yeah, for example, body weight, yeah, height, age, yeah, estate we have a like a standard, national standard Indonesia. This is the first question, and then the second, yeah, question concern about the genetic variation in SNP available in ensemble database. Are the 100 of SNP available in database, yeah, the output of specific tool such as RNA sequencing, so that access to SNP can be done quickly? Maybe Dr. Jasim can directly answer related with this question.
Yeah, so first of all, the genetic marker is using, as I told you, I don't want to give you the specific example. I tried to focus on the paper we published. So that's why I did not give those examples. But you will see there are like double muscle cattle. So they actually find the genetic marker in myostatin gene. And this myostatin gene, you know, control the muscle. So this is an example that how genetic marker are being using for selection breeding, as well as it is being using in poultry industry a lot. In poultry industry, you know, they only few people or few company control the parent stock. So they can do this marker assisted selection breeding. So I hope I give you the first answer. If you Google it, you will find a lot of examples where the marker assisted selection breeding happened for including specific SNPs. And the second question, not only the ensemble, you will also find a lot of other database like SNP DB or DB SNPs, ASEP, if you can remember. All the SNPs we identify or other people identify, we submitted there, as well as QTL DB also included all these database. So there are a lot of database. I don't want to mention one or two database. There are many database you will find. Those are publicly available. You can log in and you can find out which SNPs in which gene is contributing or associated with which phenotype, as well as if you know that this SNP in this gene is associated with a phenotype, you can then go to the ensemble and you can zoom in and you can pinpoint that specific SNP. And this is also important, like when we'll do the co-expression of the gene, because some gene help or influence the expression of other gene. This information are very, very important. So beside ensemble, you can also find some other publicly available database for SNP searching.
Okay, thank you. And Irma, I think from Irma, yeah, I think is clear, yeah, hopefully. And then let's move to the next question. So this is from Bayu. He's from, I think, from National Research Centers. Yeah, his question is, based on your experience with RNA-seq research in livestock, have any mutation been found that can significantly improve, a particular trait in breed within animal species?
That's a tricky question. If we find, that will be really game changer, but it's not like that. I can tell you that I have already mentioned you that most of the phenotype are controlled by, if not 100, at least many genes. So most of the phenotypes are controlled by many genes and many SNPs. So it is actually difficult to say that, okay, I have found out a SNP or a gene that is controlling that phenotype. So let's include that in the breeding policy. So it is difficult. If we find, we'll be very happy, but not yet. Any more question?
Okay, I think it's good. So any other question related with this? Very good. Yeah, I think experience, from you related with not only, I think, with specific commodity. Yeah, we saw you work with pig. Yeah, you work with yeah, other species like mutton or maybe sheep. Yeah, so this is yeah, maybe related with, they have any specific, yeah, regarding the commodity of species. Yeah, any limitation for using this technology or not? Because especially for species who are poor database, yeah, maybe it's little bit, I mean, difficult. For example, sheep compared to goat, yeah, how you can overcome related with this, or maybe like buffalo, yeah, camel or something like that. Maybe
That's right.
can you,
Like
yeah.
it depends on the market, you know, like we scientists are working, we need money, we need the fund. Or the industry will put money on something which will make more money. Like they are working with chicken industry, they are working with cattle to have more beef or more milk. They are working with sheep to have better wool and more meat. Or they also put a lot of money to have horse with more racing capacity, or dog with more beautiful, you know, dog. But since the company don't see the financial benefit in case of let's say goat or maybe in buffalo, so that's why those field that is still lacking behind. Lot of money in pig industry, in poultry industry, in cattle industry, but compared to that, we have very limited money in for other animal. That's why not many people are working with those species. That's why we don't see a very well database for those species. So as a scientist, I would love to have the sequence data, the genotyping data for all the species, but that's not happening. You know, this is how the business work, and we work with the industry.
Okay. Okay, so thank you. And any other question? Okay, so maybe I think because you already explained, yeah, more comprehensive. So I hope this is very good, yeah, knowledge, yeah, and we have, I think, of course, get the material, yeah, of course, from you, and then we can also distribute, yeah, to all, and then they can use, yeah, for the next, yeah, maybe project or maybe collaboration. I think it is very good opportunity, and I think it's open, yeah, from Murdoch University to open the collaboration with among us in Indonesian institution university. Okay,
Really,
so
I would
thank you,
appreciate that.
Muhammad Jasimuddin. Thank you very much for your talk, nice talk, and also with good knowledge for us, sharing with us. So maybe before I think the first session ends, so on behalf of the faculty and also department animal production and technology, yeah, faculty of animal science, yeah, we would like to give appreciation for nice talk and also deliver knowledge, yeah, related with your topic. And then maybe from our committee, we already prepare, yeah, appreciates of the certificate your talk. And then please maybe the committee can share the certificate, yeah. So the certificate of appreciation, yeah, the certificate present to Muhammad Jasimuddin, yeah, as a speaker in joint webinar for supervisor international 2000 and 2003 for postdoctoral program in IPB University. So on behalf of faculty, please accept the certificate, Dr. Jasimuddin.
Thank you.
Okay. Okay, thank you very much for nice talk. And then, yeah, I think in Murdoch, so now it's already almost 1 hour ahead, yeah?
Yeah, it's almost 4:30, but I have to go to a practical that will go on from 4:30 to 5:30.
Yeah. Okay, so thank you for your sharing. And then if you have others, maybe a teacher or maybe lecture and others, so yeah, please, or maybe I think with
Okay.
us.
Thank you very
Thank
much.
you.
It was lovely
Yeah.
to talk with you.
Thank you.
Okay.
Thank you very
Bye.
much. Yeah. Okay, for the next, maybe we have a break for Asar prayer around 10 minutes. Yeah, and then we will start again. Yeah, for the next speaker. Yeah, Professor Goran. Yeah, Anderson. Yeah, from Swedish. From Swedish. Yeah. For that, maybe Prof. Goran is already with us. Yeah, but maybe we can break first. Yeah, 10 minutes, and then we can come back again. Yeah, for the next lecture. Thank you.
I'm here. Yeah. Thank you. Do you hear me?
Yes, we can hear you, Juan.
Okay. Yes, I'm waiting for. After the break, I will be happy to give my talk. Thank you.
Yeah, thank you, Prof. Goran.
You're welcome. Okay, during the break. Do you hear me, Asep?
Hello, Prof. Goran.
Hello. Do you want me to? Hello. Do you hear me?
Yeah. Yeah.
Okay.
I can hear you very clear.
Okay. Should I try to share the screen and check that it works?
Yeah. If you can,
I
please
can do that
maybe.
now.
Yeah.
I can do that.
Brought me to supermarket to buy souvenir.
Okay. Was
Very
very nice
to
meeting
remember
you.
that.
Yeah.
Yes.
Yeah.
Okay.
Prof.
So
Goran,
please contact me and we can
Yes.
share some memories.
Yes. Thank you.
Okay.
I will.
Thank you.
Prof. Goran. Yes.
Please do. Yeah. Thank you.
So it's mean waiting for your visit to Indonesia, Prof. Goran. Many scientists.
Right.
Yeah, very
Yes.
nice to see you in this webinar. Thank you, Prof. Asep.
Yeah, thank you, Ibu Maria.
Thank you,
Okay,
Maria.
Prof. Goran, maybe we can start.
Yes, please.
Yes.
Okay.
Okay, so maybe so let's come to the second session. So it is my honors to welcome you again to the second speaker. So it's Professor Goran Anderson. Yeah, I think Professor Goran is from SLU. Yeah, Uppsala University. Yeah, like we talked before. Yeah, maybe Prof. Goran is very prominent, yeah, in Indonesia because several time, yeah, visits to Indonesia, yeah, for collaboration and also deliver also guest lecture and also to be a keynote speaker in international event like international conference from several university in Indonesia. So, and we also very, proud also because sometimes we also highlight, yeah, in our lecture, yeah, regarding your discovery, yeah, like in Buffalo, yeah, with you mentioned about the MITF gene or something like that, and sometimes we also deliver also regarding this nice collaboration. Okay, so before we give the time to Professor Goran, so maybe I want to, we would like to introduce first Prof. Sorry, Prof. Goran, so we want
Should
to
I stop
introduce.
sharing now?
Yeah.
Should I stop
Sorry.
sharing?
Yeah, I'm sorry.
Okay. Okay.
Yeah, so we want to introduce Prof. Goran. Please, the committee share the CP from Prof. Goran. So the speaker is Prof. Goran Anderson, PhD. Yeah, now faculty appointed professor at Swedish University of Agriculture Science in Sweden. They have the CV maybe from the committee that you can share, committee. before going to the topic? Yeah. So Prof. Goran regarding the education, on 1982 is BSc, Bachelor Science in Molecular Biology in Uppsala University, and then 1987 PhD in Immunogenetic Cell Research, Uppsala University, and then 1988 until 1990 postdoctoral in Molecular Genetic Transcript Channel Regulation at Howard Hughes Medical Institute and University of California. So a lot of experience, of course, but maybe we put only small size in here. So in 2009 until a current a faculty appointed a professor at Swedish University Agriculture Science in Sweden. And before 98 and 20, associate director R&D at Biotransplant in USA, and then also 1990 until 98 is scientist lecturer in Department of Cell Research in SLU. So other tasks also is a special commission in 2002, until current is a section head of molecular genetics in Department Animal Breeding and Genetic SLU. In 2019 until current is editor. This is very good journal. Is Frontier in Genetics, Frontier in Veterinary Science. So regarding for the topic now, Professor Goran will deliver the title of the topic. Maybe Komite can share what is the title. Yeah. Yeah. Professor Goran will deliver the topic related with domestic animal as a model in functional genomic and epigenome. So this is very, current topic, regarding the epigenome and also genome. So because all waiting, of course, interest to hear you, Professor Goran. So now we deliver the time is yours to you.
Okay, do you hear me?
Yeah, very clear.
Okay, so thank you for the introduction. I'm also an editor of a journal called Animal Genetics, which is I hope that you also read. It's a good journal from Wiley.
Yeah, it's very good.
Yeah. Okay, so thank you for inviting me to talk. Some of you may have heard a similar talk from me, but my research is focused on trying to understand the genetic variants that Professor Yasim Uddin so nicely described, how we identify these using the different genomic tools like RNA sequencing, genome-wide association mapping, and QTL mapping. So my research interests take advantage of all these genomic technologies that Professor Uddin so nicely described, which is good. So you understand the technologies that we're using. And I work using domestic animals then as models to understand the functional genomics and epigenomics that we study. And today I will describe two different studies. Study one is on Bos taurus, the cattle that we use in Europe and globally, of course. So it's not Bos indicus or it's not Bos javanicus. But historically, I have worked with you in Indonesia on the barley cattle, Bos javanicus. And also I have also worked as indicated earlier on the white spotted buffaloes. Okay, so the first study is on using RNA sequencing and another sequencing technology called bisulfite sequencing. and to correlate differential gene expression with differential methylation in the genome of certain cells from the cattle. The second study is one of my favorite projects, and it's the identification and function of a transcription factor called CBED6. Okay, let me see, next slide. So for some reason, okay, there. Okay, so study one has, the focus of this study is to understand the biological mechanism underlying endometritis in cattle. And as you know, it is quite common problem infecting bacteria infecting these cattle, and it causes a prolonged inflammation of the endometrium. And it is a big problem in milk producing cattle in Europe and the US. So it causes major economic losses, and therefore it is important to understand the phenotype. What is the cause of endometritis in certain cattle, and how can we overcome this by selection of individuals that are not susceptible to this. And it's basically, as Dr. Udin talked about, how we can use genomics and epigenomics to understand and then better use this information for improved selection of cattle. So we decided to focus on this by establishing a cellular in vitro model for this. And this is done then by isolating from the cow endometrial epithelial cells and treating these cells in culture in the laboratory by lipopolysaccharide, which is a surface molecule of gram-negative bacteria like E. coli, which is one of the major bacteria that causes endometritis. So establishing an in vitro model is extremely valuable because then you can reproducibly identify differentially expressed genes in response to a bacterial infection mimicked by LPS treatment. And we can also, from the same cells, isolate both RNA to do transcriptomics, RNA sequencing, and we can also do the, DNA methylation studies in response to the LPS treatment. So we correlate differential gene expression with differential methylation of the genes that are associated with the inflammatory response and of the endometrial function. Now, having established the in vitro model, we're currently working on an in vivo model using cows diagnosed with endometritis and compare them with healthy cows that are free from this disease. And this sketch here is just for you to see the principle of what we're actually studying using DNA methylation. And this is the core of epigenomics. So the ME, the methyl group, is always put on a C, a cytosine followed by a guanine. So you have a CpG dinucleotide that becomes methylated. And this methylation is critical for DNA chromatin condensation. So the condensation and the opening up is controlled by DNA methylation. We're also having the histone tails there, and the tails of the histone, the histone molecule contains a lot of positively charged amino acids like lysine, histidine, and arginine. And those are acetylated and methylated, removing the positive charge so that the negatively charged DNA can be wrapped around the histone molecules in the nucleosome. And this results in a condensation into these chromatin fibers, as you see. So we're interested in understanding then the mechanisms at the epigenome and genomic level in this in vitro model. So what we have done is that we grow these cells in tissue culture, and we have taken cells from 3 different cows. And as you see, cow 1, 2, and 3 are then treated with either 2 microgram per milliliter of lipopolysaccharide, LPS, or with 8 microgram. And this was done initially using a well-established by us concentration. The 2 microgram and the 8 microgram was established to see an effect of this. But we were not absolutely sure which one to choose, so we did both. So we treated these cells with either 2 or 8 microgram for 24 hours. And we also did, we took samples from untreated cells at 0 hour as a control. So basically what we measure in differential gene expression and differential DNA methylation is the effect of having LPS in the tissue culture media for 24 hours. And then we prepare RNA from untreated cells and from treated cells, and we compare the effect. In this case, I will show you an effect on DNA methylation. So the different, the cows are kind of clustering, as you see in this PCA plot. And we know that one of these cows actually have endometritis, pre-clinical type of endometritis. So this cow was already on its way to become sick. And upon slaughter, we go to the slaughterhouse and we take out the endometrium, and then we isolate the endometrial epithelial cells and put them into culture. And following LPS treatment, we can do this reduced representative bisulfite sequencing. And bisulfite sequencing take advantage of the fact that when you treat the DNA with bisulfite, it discriminates between CpG dinucleotides that are methylated versus those that are not methylated. So we can observe differential DNA methylation in response to LPS treatment. And in panel B there, you see that there is a large number of differentially methylated regions. We call them DMRs. And there is a total of more than 1,200 different differentially methylated regions. Now, since we have the genome sequence, the reference genome sequence, and we have the RRBS, the bisulfite data, we know exactly where these differentially methylated regions are. And we have shown that the majority of these, if you go into the next slide here, you can see the distribution in panel A. So these are all the chromosomes of Bos taurus. So it has 29 autosomes. And then these are cows, so we have also the X chromosome. And you can observe that on chromosome 1, which is the longest of the autosomes, there are relatively few, all these dots here, you can see the positions of the DMRs as a little small bar. So there is an uneven distribution, but the take-home message is that the differentially methylated regions are distributed on all the chromosomes in the cow. There is a certain enrichment, actually, at the ends of the chromosomes, indicated by the red and the yellow. So the telomeric regions are actually enriched for differentially methylated regions. So that's an interesting finding. We know from before that telomeric regions are also enriched for genes that are imprinted, meaning imprinting means that it's only one of the two alleles that is expressed. And I will come back to imprinting in my second, the study number 2 that I will describe. So evenly distributed, some chromosomes have fewer and some have more, obviously. You can see the density there. And also an enrichment towards the telomeric region. Then we can look at panel D also, where we have actually taken a look at particularly where are these DMRs, the differentially methylated regions. And they are enriched in what we call CPG islands. So islands that are particularly rich in CPGs. And those are known to be regulatory DNA sequences like promoters, enhancers, locus control regions that are critical for controlling transcription of genes. So what we do is that we look at the differentially expressed genes that we have identified using RNA sequencing, the method that Professor Udin described. And then we're taking a look at if a gene is activated or if a gene is downregulated. We correlate that with its nature of its status of differential DNA methylation. So we can show that there is a particular correlation between differential gene expression and differential DNA methylation. And that in all these individuals, all the individuals DMR, the majority are located in promoters and enhancers. We also show, I mean, this is an example how you can present your data where you integrate the differential gene expression data with the differential DNA methylation data, and you see a nice we see a nice correlation. As you may know, is that if a promoter is demethylated, we call it hypomethylated. This is correlated with active transcription. And this is actually perfectly what we see. We see that genes that where the LPS induce transcription, we see that that particular promoter is reduced in DNA methylation. So there's a very nice correlation between hypomethylated promoters and activated transcription of a set of genes in these cells. And which are these genes? Udin also showed gene ontology data and pathway analysis. And this is what we, when we analyze our differential gene expression data and our differential DNA methylation data, the conclusion is that there is a massive expression change correlated with a massive differential DNA methylation. And the genes that are influenced by the bacterial infection, the in vitro model for bacterial infection, are predominantly, as expected, maybe you can think, involved in inflammatory responses. But it's a confirmation that this in vitro model is actually mimicking an in vivo endometritis. So it's a very useful tool for us to move further into the in vivo studies. And the biological processes you can read are both involved in inflammatory responses, but importantly, also in implantation of the embryo in the endometrium. So what we can observe from these in vitro studies is that when the cow experiences a bacterial infection, it leads to inflammation, and this inflammation negatively influence the implantation capacity of a fetus into the endometrium. And this is, of course, not shown in our in vitro model, but it's the conclusion when we look at the differential gene expression. These are genes that are well known to be critical for implantation. And in panel C here, we show the hub of the network, and there are some critical nodes, the MAP kinase signaling, calcium regulation, and also some disease susceptibility pathways that are indicated as being nodes of these strong interactions of genes that we see. So the conclusion from this study is that LPS, lipopolysaccharide, which is called also endotoxin from E. coli, massively altered DNA methylation pattern in these bovine endometrial epithelial cells. The information combined with the changes of gene expression confirmed that LPS activates these pro-inflammatory mechanisms, leading to a perturbed immune balance and cell adhesion processes at the time of recognition of pregnancy. So cows that are influenced by this, they have reduced fertility because it negatively influences implantation of the embryo. So this in vitro model for endometritis, we have identified a large number of differentially methylated regions in promoters and enhancers of genes that are critical for immune regulation and implantation. We have a number of references that I can, you don't need to write it down. I can make this presentation available to the committee so that you can take a look at them. But it has been a number of interesting papers from this in vitro model, and we're currently moving into an in vivo model where we will take endometrial cells from cows that have been diagnosed with endometritis and compare them with healthy cows. This current project is funded by an EU project. It's an EU project called Rumigen. Okay. The next, actually, this is a slide taking you to the next study. And actually was someone from before the talk before, that someone called Bayou, of some mutations, gene variants that have been shown to significantly improve a trait. And I have actually a slide of that. There are a number of them. Myostatin was mentioned by Dr. Udin, MSTN. It's a double muscling, it's a loss of function mutation, both in cattle and sheep. The DGAT1 gene is a success story in dairy cattle, causing massive effects on milk yield. So, but there are few cases where we have gone from association using quantitative trait locus QTL methods or GWAS method, where we have actually come all the way to identifying the causative mutation. And today I will describe a project that I have been involved in calls in the IGF-2 gene in pigs, influencing postnatal increase in muscle. And IGF-2 is the insulin-like growth factor 2. It's a very important growth factor for all cells in our body. And it's also one of the most important embryonic growth factors. I have also been involved in the MYH3 study, also in pigs, and it influenced intramuscular fat content, marbling of muscle. So noteworthy of all these studies is that the mutations that are causative influences transcription of the genes. So they are present in enhancers and promoters of these genes, and they are changing the capacity for epigenetic modifications. And the figure to the right there shows schematically how the mechanism for the IGF-2 mutation, which I will describe to you in some detail now. So it started more than 20 years ago by my colleague Leif Andersson did an intercross between a wild boar male to the left and a domestic large white sow to the right, and producing a large number of F1 litters that were then crossed to each other, F1 cross, producing more than 200 F2 individuals. So using this experimental approach, you have sort of a gold mine for a geneticist to map genes controlling various different traits. And in the trait shown in this figure here, it's coat color. So the individual in the middle there has the wild boar coat color, whereas the one to the far right has the domestic large white coat color. And the white coat color in pigs is called dominant white. So there is a segregation in the F2 of these, whereas all F1 individuals are white, dominant white. So what an intercross takes advantage of is that we can assume that all the domestic large white are homozygous for all the mutations that have been selected for hundreds of generations, thousands of generations, I should say, for many, many generations. So they are basically undergone a selective sweep of all the causative mutations that we have selected for. And as you know, in pigs, we have selected for meat production. So all genes influencing muscle growth have been selected for in the domestic pigs, whereas the wild boar to the left, the wild individuals, they are wild type homozygous for all the muscle genes that are required for a functional life in the wild. So they are homozygous for all the good genes that a wild boar needs to survive in the nature. Okay, so this is a very powerful way, and what we did here was to measure the phenotype of muscle growth in the F2 generation. And we did a so-called quantitative trait locus mapping. And it's called a QTL. We knew that the particular trait that we were after was a paternally expressed QTL. And it affected both skeletal and cardiac muscle. And a paternally specific expression is what we call imprinting. where only the allele we inherit from our father is expressed, whereas the maternal, the one we have from mother, is silent. So this is the background of this particular QTL is that the phenotype is paternally expressed. And then you do a genotyping. In this case, it was done using microsatellites, which are highly polymorphic genetic markers. If we would have done it today, we would have used one of the SNP arrays that Professor Udin described to you, like 100K porcine SNP array. But this was done before these arrays were available, and it was using highly polymorphic microsatellites scattered on all the different pig chromosomes. And using this approach, we could identify a very potent QTL on the telomere of pig chromosome 2. There is a small mistake here. It should be 0.5% in the middle there. So anything above the 0.1% level is statistically significantly different between pigs with or without this muscle affecting genetic variant. So as you see, lean meat in ham and lean meat mass in ham is strongly associated with increased muscle. And it maps to a position where there is only one gene exhibiting this imprinted phenotype being expressed from only the paternal allele. And this is the IGF-2 gene. And the IGF-2 QTL allele from domestic large white increases muscle content with some between 3 and 4%, together with its reducing subcutaneous fat and also an increase of the heart. So this is a very nice data for us, was published now 20 years ago, so it's old, but it's still heavily cited. So this paper has been cited many, many times. And it's a sort of a success story on how you can, using domestic animals, identify very potent gene variants influencing, in this case, muscle growth. And it has, there was another question actually during the previous talk, whether one has used this CRISPR technology to introduce some mutation. And this particular mutation, which I will describe how we identify it, has been introduced using this technology in other populations. So always in science, it's good that if colleagues from other laboratories can corroborate the finding that we have. And we used the wild boar large white intercross. Colleagues in Belgium use the P-Tran and large white. And then also Meishan in another group, and also Hampshire. And they all confirmed that the association is on pig chromosome 2, overlapping the IGF-2 locus. So in order to identify, you know that in a QTL approach, you identify a region of association defined by the extreme microsatellite alleles. And we assume something in this technology, that the mutation that influenced this phenotype is identical by descent, so that all individual pigs with the phenotype share exactly the same mutation. And this is called IBD mapping. So what you do is you identify the smallest shared haplotype among the chromosomes associated with the mutant allele. And this is a sketch that I will show you. By definition, all these bars here are chromosome, and the red chromosome is the one affected by a mutation. Changing, we call it small q goes to big Q. And by definition, from day one, in a population of different chromosomes, in a population of pigs, it is a single mutation causing an effect, and this is the big Q mutation. Now, it is from day one, it's in linkage disequilibrium with all other variants on that chromosome, but over a number of generations, n generations, there are recombinations between that chromosome and the wild type chromosome, and it happens at random in all individuals, and we can then define the minimum shared haplotype containing the mutation. And this is by the second to the right, and the one in the middle is the region. If we display this in a relatively complicated slide, but I will walk you through it. So what we first did is that we resequenced 15 chromosomes classified as either big Q or small q from individuals phenotypically classified as having the mutation big Q or being wild type small q. And among the region that we defined is a relatively big region still, has many potential gene variants that could be the causative variants, but it spans from to the left there, the tyrosine hydroxylase gene, which is not expressed in muscle, so that one could, not be the causative gene. Insulin is actually potentially, the next gene there, insulin, is potentially influencing this because it is imprinted in embryonic yolk. But the major causative gene is the IGF-2 locus. And on this upper panel here, all these yellow boxes are CPG islands, and the red boxes are exons of the IGF-2 locus. And you see also P1 close to the insulin is one of 4 different IGF-2 promoters. And P1 promoter is only active in the embryo, in the placenta. P2, P3, and P4 are expressed in the adult organism, like you and me. We have also these different, humans have also these different promoters, and P2, 3, and 4 are expressed in different adult cells. In the case of IGF-2 and muscle, it's P3 that is the most important promoter. So when we resequence this, we identified a set of SNPs shown by these small red, blue, and green bars. And the one in IGF-2 intron 1 defined the left SNP of the minimal shared haplotype, and the one in overlapping exon 9 in IGF-2 is the one to the right. And as you see, we were lucky because one SNP immediately identified the causative mutation, and it is in intron 3 of IGF-2, overlapping a CPG island, and it is indicated by this star there. And a single nucleotide mutation, changing a G in a CPG dinucleotide to an A, And G to A transitions are the most common type of mutations that we can observe in a mammalian genome. And the CPG there in intron 3 in the wild type is one of these CPG dinucleotides where the C is methylated. So we immediately understood that it could have something to do with changing the methylation status, because you remove by the G to A transition a CPG, it becomes a CPA instead. And we call this a QTN, a quantitative trait nucleotide instead. And actually we did, this was some years ago, but we actually still do northern blotting, even though it's time consuming, very tedious, requires a lot of RNA. But the important point of, and then we use also, as Udin talked about, RTP, real-time PCR. But the beauty of using northern blotting is that you also detect all the sizes of the transcript of the gene in question that you're interested in. And this is what we did. We prepared RNA from individuals' phenotypes being a big Q versus a small q, and then we loaded RNA on these northern blotting gels. And as a housekeeping gene, we used GAPDH, and you see that we actually loaded more RNA in the small q individuals. So the effect that you can observe is a quantitative effect in the big Q individuals, expressing more transcript of IGF-2 initiated from the P3 promoter, P3B there. And this is also shown using the effect on the RNA level is also shown in the panel RT-PCR. We look at fetal expression. There were equal amounts in the small q and big Q, but after birth, from 3 weeks old pigs all the way up to 6 months old, the amount of IGF-2 mRNA was statistically significantly increased in the muscle, in the skeletal muscle of these pigs, all the way up to 6 months. The decrease from fetal to adult or after birth is well known for IGF-2. It goes down tremendously because it's a major fetal growth factor, but then in an adult muscle, for instance, it goes down until the muscle experiences hypoxia. Then hypoxia, low oxygen level in the muscle, increases transcription of IGF-2. So it's an hypoxia-inducible gene. The effect is also observable in 3-week-old heart muscle. But the main producer of IGF-2 in an adult organism, including humans and pigs, is the liver. And it is only slightly more of IGF-2 in, but not significant. So the liver effect is not influenced by this QTN. So the QTN is in a enhancer controlling transcription from a muscle in a muscle specific manner. So this is an important take-home message. And when we have this kind of information, a mutation influencing transcription, the immediate hypothesis is that a transcription factor is perturbed in its ability to bind the DNA when you mutate a highly conserved nucleotide in a enhancer of, in this case, the IGF-2 gene. So then we do some biochemistry and we prepare the sequence from the small q versus the big Q DNA, and we test that DNA, radioactively labeled DNA, for its ability in vitro to bind nuclear proteins. So you can prepare nuclear proteins from cells grown in culture. And these nuclear proteins are then, of course, able to bind DNA because they are transcription factors predominantly. And you can then compare their ability to bind to wild type versus mutant. And here we got a very, very nice result. In lane 1, you saw the wild type can bind 3 complexes, C1, 2, and 3. When you compete with increased amount, 10 molar excess of the cold small q probe, you compete out C1 completely, more or less. I mean, there is only some minor left there. But when you compete with the mutant, you do not compete. And when you labeled the mutant, it doesn't bind any transcription factor. So it's an absence of C1. So C1 is a complex between the wild type DNA and an unknown protein present in myoblast nuclear protein extracts. And then we spent several years actually identifying which is this protein, because electrophoretic mobility shift analysis, EMSA, doesn't tell you which protein it is there. Could be more than one protein also, but it's a biochemical method showing whether or not there is something binding to that particular DNA or not. So what is this protein? Wild boar sequence to the bottom there, you see that the CPG has been changed to a CPA, and it is only the small q, the wild boar sequence, that can bind to this. And the protein we worked hard to find, and there was a method developed by 2 groups, one in Germany and one in Boston, in Massachusetts, in the US, and we collaborated with the group in US, and it's called SILAC, Stable Isotope Labeling with Amino Acids in Cell Culture. So we use the same cells, C2C, C12 that we used for the EMSA, and we allow these cells to grow in 2 types of radioactive amino acids, with natural isotopes in red to the left, and in a separate growing cells using heavy amino acids. So heavy versus light can then be discriminated because these amino acids are incorporated into the proteins grown in these cells. And they can then be isolated using a bead association, biotin streptavidin technology. So the beads are loaded with DNA, with either the mutated DNA sequence or the wild type sequence. And remember from the previous slide that it's only the, I can go back to that, it is only the mutated is from the big Q, and the wild type is from the wild boar. So in the in vitro data, it is only the small q that will bind. So we do, then after binding, we separate the proteins that do not bind, and we isolate the protein that has bound to the bead with the wild type protein then. And then we separate them, wild boar versus large white, on liquid chromatography. And then you isolate the protein, you digest the protein, and you sequence the protein. And we detected a amino acid sequence from one of these peptides that strongly was discriminative in its binding, differential binding only to the wild boar sequence, the small q, and background only to the big Q. And this particular sequence is shown here, the peptide that we have here. And this peptide sequence was then mined in publicly available databases. And it was not found easily, but it was present in the human genome supplementary files of an unknown open reading frame that we could show was very similar to a DNA transposon. And DNA transposons are the jumping genes that we all have in our genomes. And they were identified by Barbara McClintock using corn, popcorn, Sia mice, many years ago. And she got the Nobel Prize for this. She didn't know what the DNA transposon was, but she could observe that there were some DNA sequences that were moving in the genome of these plants. And when we found this, that the sequence of this enriched protein using the Cy-like method was similar to a DNA transposon, we thought it was a mistake, right? We were a little bit depressed for a while there. But then we realized when we looked into the literature that there were a small number of DNA transposons that had been domesticated during evolution. And these domesticated DNA transposons were grouped into different classes. And our sequence was the member number 6 of a small group of 5, now it's 6, domesticated DNA transposons. And they are then transcription factors controlling expression of very important genes. The founding member of this small family is called CBED1. It controls transcription of the gene encoding DNA polymerase 1, which is the enzyme doing replication during every time a cell divides. So it's a very fundamental usage of DNA transposons to control transcription of eukaryotic genes. And the DNA transposons, as you may remember from your biology studies, they are moving using their own proteins by a cut and paste mechanism. So they move around, and in order for their ability to cut out themselves and insert themselves, they need to have a DNA binding protein expressed by themselves. And this DNA binding protein is a zinc finger protein. And zinc finger proteins represent the largest class of eukaryotic transcription factors. And this, in the case of the CBED6 protein, it has 2 zinc finger boundary element domains, these red boxes there. CBED6, the protein we are interested in now, also have a dimerization domain. And the names comes from DNA transposons from insects, Drosophila, from plants, maize in this case, and also from the snapdragon, which is a plant. And CBED6, tremendously conserved protein using a cluster analysis. The different versions of the CBED family cluster in different families of DNA transposons, and CBED6 is in the AC family. For those of you that might be interested, you should read this paper by us, published 10 years ago in PLOS ONE. But as you see, there is a huge number of DNA transposons in our genomes. And we are only starting to appreciate that some have been domesticated, meaning that their activity to move around has been prevented. And this is exactly what has happened with CBED6. It has integrated in intron 1 of a host gene called ZC3H11A, it should be there, down there. And it sits downstream of transcription start site of this host gene. And it is produced by alternative splicing. So if intron 1 is spliced out at the mRNA level, the host gene mRNA is being translated. But if the exon, if the intron is retained, CBED6 will be produced. So intron retention of intron 1, CBED6 protein is produced. Normal intron splicing, the host gene is produced. Host gene requires a separate talk, actually. It's also extremely interesting protein. Now, looking at the DNA binding, the boundary element domain of this CBED6, it is 100% conserved among mammals. And now with the Sonomia project being published in Science a few weeks ago, they have sequenced 240 different mammals. We can increase our study to see if CBED6 is 100% conserved in all these 240 mammals. We haven't done that yet, but that will be done. Now, looking at when did this happen, it's present in all eutherian mammals, but it's also present in the monotreme, the platypus, and it's also present in marsupials, opossum and kangaroos, etc. And the conclusion from this is that CBED6 was domesticated in a primitive mammal more than 200 million years ago. And the split between the monotreme and the others occurred more than 150 million years. So of course we have a span there of a long time. We don't know exactly when this integration and domestication happened, but it was a very large number of million years ago. More than 180, but we usually say more than 200 million years to be certain. Then in the marsupials and in the monotreme, the protein has, the gene encoding this CBED6 has mutation preventing it from being expressed as a functional protein. But in all eutherian mammals, including humans, here we are, humans down to the right there, it is a functional protein expressed in many, many different cell types. So now we wanted to do functional studies to define how this point mutation in the intron of 2 of IGF-2 is influencing transcription. So we took advantage of the same type of cells again that we have used before, C2C12, and we performed something called a luciferase assay. And briefly, this takes advantage of luciferase, which is a reporter gene not expressed in mammals. It is from the firefly or from a marine animal. And activity of the luciferase is then controlled by what you put into the vector controlling transcription of the luciferase gene. So in our case, we put in the P3 promoter. Remember, the P3 promoter is the one promoter that responds to this mutation. And we also put in the next construct the actual wild type small q enhancer in front of the P3 promoter. And then the bottom one, we put in the mutant. And then we transiently transfected these vectors into the muscle cells, and then we measured luciferase activity. And the P3 promoter alone is the one activity we set as a comparative comparison to the wild type with CBED6 binding activity and the big Q without its binding activity. And you can see that the wild type, the one from the wild boar, functions as a very potent transcriptional repressor of the luciferase activity. So whereas the mutant, the one from the large white individual, is not a repressor. So the single base pair mutation removes a repressor activity required to control transcription of IGF-2. So CBED6 functions as a transcriptional repressor. This is the hypothesis from this data. And we moved on to confirm that it is caused by CBED6 alone. And we took advantage of a technology called small interfering RNA. So by introducing this in the tissue culture cells used for the luciferase assay, the siRNA will remove the mRNA for CBED6 specifically. And the P3 promoter, after siRNA has been applied, removes this repressor activity. Remember, this went down to close to 5 here, from 30 to 5. But when we added siRNA, these were then back as high as the P3 promoter. So siRNA against CBED6 is in the luciferase assay provides conclusive evidence that this is the nuclear factor interacting with the CpG island. And it's a transcription factor functional as a repressor. It is a transcription factor then that repress IGF-2, and it binds to its site on this CpG island in a methylation-dependent manner. And we have done something called chromatin immunoprecipitation sequencing, SHIP sequencing, using an antibody developed in our lab against specific for CBED6. And using that antibody, we can identify all the sites that CBED6 can bind to in the genome. And it revealed 2,500 different sites. Our positive control is the Q10 region in IGF-2. And the derived consensus motif for all these 2,500 sites is perfectly corroborating the GC-TCG motif found in the small q sequence on top of in panel B there, whereas the mutant changes one of the critical 5 nucleotides, a G to an A there. We know that in vitro it is binding DNA in a DNA methylation sensitive manner as well. I was surprised by the large number of sites, but this is kind of typical for a potent transcription factor. It binds to many, many places. The challenge we have now is to confirm, and that has been done by several other papers, that CBED6 also functions as a transcription factor for many, many other genes besides IGF-2. Now, we were interested in looking at these SHIP-seq data, chromatin immunoprecipitation using the CBED6 antibody. Where are all these 2,500 sites? Obviously, in promoters, enhancers, but we also wanted to define the histone code at those particular sites. And then we could use commercially available antibodies that recognizes different modifications of histones. So on top there, the red one, H3K4ME2, means that it's K is for lysine, H3 is for histone 3, and Me2 is that it's dimethylated. So histone 3, lysine number 4 in the tail is methylated twice in promoters that are actively transcribed. This is a well-known epigenomic feature. Enhanced, and there are a couple of others, H3K4 3-methylated, and K27, another lysine, when it becomes acetylated, it is correlated with active promoters. And you can see that the two active promoter marks, histone marks we call them, are then strongly enriched in CBED6 sites. So what it means is a transcriptional repressor binding to promoters that have been primed to become activated when the signal comes. And this is something that is of profound importance for our understanding, because in this schematic that you had already in the beginning of my presentation, you see the wild boar has only a small amount of IGF-2 being expressed, and the protein is binding to that CPG indicated there. But IGF-2 has to be activated very profoundly when the individual muscle starts to work. Because as you know, if you go to the gym, or if you start running or whatever exercise you perform, you develop fatigue and you develop lactic acid in your muscle. And this is something caused by the hypoxia in your muscle. And as maybe you remember, I told you in the beginning that IGF-2 transcription in muscle, in skeletal muscle, is hypoxia inducible. So the chromatin structure at the IGF-2 locus in muscle is in open chromatin, and the transcription factors needed to induce transcription is waiting for the signal, hypoxia. And the hypoxia then releases the repressor and increase transcription. Now, if this repressor cannot bind, you have more transcription of IGF-2 that cannot be reduced by the CBED6 repressor, shown here in the Hampshire and the Pietran. So it is a powerful powerful transcription factor controlling inducible transcription of IGF-2 and increasing muscle weight when it is mutated here by 3 to 4%. It may sound small to you, but consider it being enriched to 100% in pig breeds used for meat production, and 3 to 4% more muscle in every single pig. This mutation is one of the most economically important mutations that have been selected for by, remember, traditional breeding, not master-assisted breeding or anything. But now, currently, it is used also by scientists to do that. So sort of a model for CBED6 function here is that when it binds DNA, it's a repressed state. If you mutate the site, you become active to the left down there. If you methylate the site, CBED6 cannot bind, and that's an epigenetic activation of transcription in genes controlled by CBED6. You can, of course, also downregulate transcription of CBED6 by other means at its promoter and enhancer. But in some cases, you can have loss of function also, so that you don't produce the CBED6. This is the model for CBED6 function, and the conclusions from these studies was when we published this, it was the first ever single point mutation identified as a regulatory mutation underlying a quantitative trait locus. So it's a QTN. And this work illustrates the extraordinary resolution that is achievable in the molecular dissection of complex traits using domestic animals. And there is a large number of publications for this. The one identification of the QTN was published 2003. The IGF-2 antisense transcript that I haven't talked about today is also regulated by this region. And then the identification of CBED6 as a novel transcription factor was published 2009. This in transcription here is a review article. These are all publicly available papers, open access. The Hayward paper showed the evolutionary relatedness of the seabed family. I should say that there is a paper that I haven't listed here by a colleague of mine in Uppsala, whereby they have looked at where is there an enrichment of seabed 6 binding site. And very interestingly, it is in retroviral LTR. Our genome in mammals consists to something like between 8 and 10% of endogenous retroviruses. And endogenous retroviruses are controlled by long terminal repeat, the transcription. So LTRs are also domesticated. So a quarter, 25% of all promoters and enhancers in our genome is originated from endogenous retrovirus LTR sequences. And the paper by my colleagues, they showed that seabed 6 binding are enriched in LTRs derived from endogenous retroviruses. So there is an interaction between DNA transposons, jumping genes that move through a cut and paste mechanism, with endogenous retroviruses that move with a copy and paste. So they increase in copy number. During evolution, we have taken advantage of the tremendous specificity of retroviral infectivity. And if you don't appreciate what I said just now, you can think of HIV, which is a retrovirus that it has yet to be shown as an endogenous retrovirus, but it is a retrovirus that specifically infect T helper cells and macrophages, because it is a tissue, retroviruses have tissue tropism. So the recruitment of LTR promoter sequences are usually very important for tissue-specific transcriptional control, because the LTRs infecting a T cell have the transcription factor binding sites for transcription factors present in the T cell. And this is true for the HIV LTR, for instance. Okay, so there is a lot of work going on seabed 6, and I have not listed all of them here, but you can Google yourself if you want to read more about it. Acknowledgements for these studies. Erik Bonkam Rudloff is a professor in my department. He's a bioinformatician. Adnan Niazi and Navid Jamat are also bioinformaticians. They were involved in the study number 1, using the RNA sequencing and the bisulfite sequencing analysis. Maria Wilbe was also involved in that study. And the study was done in collaboration with Professor Patrice Humblot and a postdoc in his lab called Zhongxi Gu. The study by seabed 6 is done in collaboration with Professor Leif Andersson and Professor Kerstin Lindblad too. And if you haven't heard about these guys, they are world-leading geneticists. And Kerstin's group published 11 papers in Science a couple of weeks ago, or if it was last week. No, it was a couple of weeks ago. In this is called the Sonomia project, where they have sequenced 240 mammals. It's a tremendous resource for all of us interested in genomics and epigenomics. So you should take a look at that. And thank you for your attention. And there are a couple of, I've listed also some key papers that I have talked about during my talk here. So thank you for your attention, and I'm open for questions.
Yes, thank you very much, Professor Goran. this very nice talk and also very wonderful work. Yeah, it's a lot of work, of course, and also long experience, but we saw this a
Right.
lot of published paper in very high reputation journals like Nature and others that maybe we dream come to publish in this published paper like that. So it's very interesting, and then we saw characterized is very deeply and cause mutation like EGF2, and also in the last is GBAD6. Yeah, and then we hear it's very comprehensive study.
Yeah, exactly. Right.
Yeah. Okay, so maybe now, of course, many from all of the participants want to know, yeah, more. And then now we open the question and answer. Maybe so if somebody or maybe everyone who want to ask directly to Professor Goran, maybe before I read from the chat, maybe I give opportunity first to all the participants. Somebody want to ask directly to Professor Goran? Okay, so maybe
I can take
during
a question from Irma.
Yeah.
The myostatin gene in cows. It's
Yes.
the first one was okay. If we cross the cow with a local cow that produces heterozygous offspring with normal appearance, is there still a potential for weight gain from other factors such as the effect of heterosis due to differences in breed? How do you think the difference is increasing muscle mass between the effects of heterosis? Right. So I think that it is a very good question. The myostatin mutation was identified first in a breed called Belgian Blue, and it is a loss of function mutation. So it inactivates the gene, and myostatin is a member of the TGF beta family, and it's a very potent repressor of muscle growth. So when you have a loss of function mutation, you will then increase muscle, and in homozygous version, as far as I know, it is double muscling. And there is also, when you look at a large number of domestic cattle breeds in Europe that are used for meat production, you have various different mutations in the myostatin gene. So the Belgian Blue mutation is a complete loss of function, whereas the other ones are more subtle, but they are still very potent. So I would not recommend to introduce a loss of function in your cross. I would use a reduced function so that you don't get the massive phenotype of the Belgian Blue, because these cows, they're forbidden to breed in Sweden, by the way, because they cannot be delivering their calf in a normal way. They have to be cesarean section, I think. But there are a number of other beef-producing cattle breeds where you have missense mutations. You have amino acid substitutions that causes increased muscle weight. And I would, if you want to do a cross like that, I think you should do it. I think that what will happen is that in heterozygosity, there will be a less profound phenotype, and in homozygosity, you will have a more massive effect of the mutation. I'm not sure. I think you have to detect, the literature, which, if you're planning to use this, which one you would like to cross with. I would do perhaps some mutant, and there are review articles where you can see all the different mutations that have been described. There are more than, I don't remember exactly the number, but there is a large number of known mutations in the myostatin gene. You can, I mean, as Udin talked about, you can introduce this mutation if it is known, if it is not known, if it is okay to do that regarding your legislation. We're not allowed to do it in Sweden. But the CBED6, the QTN, has been introduced in China by colleagues there. So by CRISPR-Cas technology, you can also introduce a mutation in an individual. Of course, this is a possibility if it is legally acceptable in your country. I don't know. It is not acceptable yet in Sweden. And the reason for that, I may mention actually, is that the use of CRISPR-Cas is a fantastic technology, right? You can introduce the mutation, but it is not free from off-target effects. So it means that the technology is not specific yet. So when you introduce the mutation in your target gene, you cannot exclude that the mutation has been introduced in other places in the genome as well. So this is a major problem, and this is the main reason why it's not acceptable to do in Sweden and in many other countries. So the off-target effect is still a problem. I know that a massive effort to use other Cas proteins, you have CRISPR-Cas9, but there are CRISPR-Cas2, 3, 4, 5, etc. So there are many other Cas proteins that they hope to be less, even more specific. Okay, is that, did I answer your question? Irma?
My question about the second question, how do you think the difference in increasing muscle between the effect of heterosis? Because we use two different breed, Belgian blue and Bos Indicus, such as small grade in Indonesia.
I think the question is a classical one. I mean, in all individuals, the effect of a mutation will be influenced by the genetic background of that individual. And of course, there may be compensatory mutations counteracting the effect of the mutation that you introduce. And it will be different in different scenarios. But to the best of my knowledge, at least in the Belgian blue, there is a complete penetrance of the mutation in homozygosity. And I'm not an expert on all these other mutations in the myostatin, but I think that you have to consider that, something like copy number variation is of course important. Many genes that are, myostatin is massively activating a lot of genes together influencing muscle growth. And myostatin is a signaling molecule binding to some kind of tyrosine kinase receptor. And this signaling is then thousands of genes may be activated by this. And of course, the genetic background of that, when you compare two different breeds, will differ in the genetic background. So the same mutation will have different penetrance in different domestic animal breeds. Okay. I hope I answered your question. Is it okay?
Is it okay, Irma, to answer your question?
Thank you.
Okay, thank you.
Okay, thank you.
Prof. Goran. And they have also other question from Professor Endang, Sri Margawati.
Okay.
Prof. Goran, is his spot color and albinism is different? If yes, then are they affected by different genes?
Okay, the question was albinism is a is a mutation preventing melanin to be produced. And it is, you know, in the pathway of being an albino, in the pathway of this, it is a converting enzyme changing a tyrosine to a pre-melanin form. And if you have a mutation in that enzyme, in the gene encoding that enzyme, you cannot produce melanin at all. Then if you look at coat color mutations causing phenotypic coat color changes, the species we know most of is the mouse. And the last time I looked at the website of the site in the US, which I've forgotten about now, there are more than 150 mutations influencing coat color. And many of them will influence whether you get the migration of melanocytes. So the MITF mutation that I have worked with, it is influencing the migration and development of melanocytes, but it doesn't prevent melanin production. So that's why you get the spotted phenotype of many MITF mutations. So albino, the difference between albino and most of the other is that the albino doesn't produce melanin whatsoever. It's completely avoiding, eliminating melanin production. Whereas most of the other that I know of reduces or changes the expression amount of melanin. And you have, of course, eumelanin versus pheomelanin, the reddish phe versus the blackish eumelanin, etc. And then you have a large number of genes that can influence coat color. And again, as I said, there are more in the mouse, there are a large number, probably close to 200 different mutations known that influence coat color. Interestingly, coat color is very often a pleiotropic phenotype. So besides influencing coat color, mutation also influences other phenotypes. Is that okay, Endang? Okay. I don't know what happened now.
Thank you. Thank you.
The cord ASEP disappeared. So
yeah.
So are there more questions?
No, I couldn't see.
Okay, Jafar, I want to ask, what are the key challenges and ethical considerations associated with using domestic animals as models? Well, and how can these concerns be effectively addressed? Well, I can tell you that we have very strict ethical considerations associated with our use of domestic animals. And in the case of cattle that I have described to you, and in the case of pigs, we have, before we can do any experiments whatsoever, we have to apply for an ethical permit to do these studies. And the ethical permission is given by experts in both animal health and behavior, animal health and welfare, and also legislation people. And the criteria for getting this permission are quite stringent in Sweden. And as an example, in the in vitro studies in study 1 that I described, we got the permission to take out endometrium from slaughtered cows that were slaughtered as a planned slaughter for milk producing cows. So they were not slaughtered because we wanted to take out an organ. They were slaughtered for other reasons, for production reasons, basically. In the work of the pigs, we got a very stringent ethical permit to actually breed these animals and taking samples of them when they were slaughtered for also other purposes, for meat production purposes. So in Sweden, pig production for meat is very important, and these animals were not, they were slaughtered and measured in a manner that prevented any distress besides being euthanized. Of course, it's a distress, but they were euthanized for other reasons. In our other work on companion animals, horses, cats, and dogs, which we also work a lot with, the permission to take a blood sample, which is the most commonly sample we take, is given by ethical permission by the owner of the animal, and the permission to use it in research only, and it is also anonymous, so that no one reading a publication coming from our studies can identify the sample, the individuals that provided the sample, or the owner providing the permission. So it's a completely blind thing, and the only in my research, we try always to do non-invasive sampling, and the most invasive sampling that I have in a live animal that I have been involved in is skin biopsies. So it's a relatively small invasive. I mean, it's I have never taken a skin biopsy myself, but it's relatively painless and it's not invasive. And so can these concerns be accepted to ensure responsibility in the scientific? Yeah, I think that we always highlight the importance of animal welfare, and we're not using these animals to. In contrast, we're performing our studies to improve health and welfare of these domestic animals. What specific function? Okay, so well, the last question is really good. We are always using a comparative aspect of our studies. So the evolutionary similarity in many of these gene systems that we work on. So what, for instance, what controls muscle growth in pigs is the same system that controls muscle growth in humans. And one of the aspects, as an example, the work that we have done on the CBED6 and IGF-2 can have profound importance in understanding conditions in humans when we grow old, or when we become sick by, we have to lie down in the bed all the time, because that is coupled with muscle wasting, so a reduction of muscle muscle mass, and our insights in how muscle mass is regulated is, of course, of comparative value to understand what controls muscle wasting in humans. as a comparative value for that. We actually thought about, in the beginning when we identified CBED6, before we understood that it's important in not only muscle but in brain also, for instance. But you know, modulating the activity of a muscle effect of CBED6 is of course of potential value, and we were actually planning even to submit an application to NASA, NASA, which is the Air and Space Organization in the US, because what is the problem for astronauts is muscle wasting when they're up in the space. But we realized that we haven't done it yet. But basically, the use of domestic animals is a wonderful way of learning more about complex biology in mammals in general, and also in birds, by the way, because we of course do studies also in chicken. I haven't done a lot of that myself, but my colleagues are working a lot with chicken. And in the case of the dog, we use the dog as a model for monogenic and complex diseases. And these are always, you know, as I described to you earlier, dogs that have, where the owners have come to the clinic seeking help for veterinary care to treat the dog for whatever disease it may have. So as you may know, there are more than 400 different dog breeds, and each dog breed in Europe. And I remember a funny story actually. I asked Ronnie Noor the first time I was in Indonesia, when I was working with dogs a lot, and I saw some dogs there, and I asked Ronnie what kind of breed it was, and there is no breed like we have. I mean, it's just a street dog or something like that. So they are breeding independent of humans, basically, which is not true in Sweden. I mean, you never see a stray dog in Sweden. They're all privately owned dogs, and the breed is defined by strict criteria for which dogs you can use for breeding. The problem with that is that it increases inbreeding. You lose genetic variation, and the loss of genetic variation, an increase in disadvantageous mutations, leading to increased risk of developing a large number of diseases. But importantly, these diseases, the clinical phenotype of the diseases are extremely similar to diseases that we develop. So I work primarily with immune-mediated diseases like autoimmune disease, atopic disease, and there are dog models for the diseases that we work with. So for instance, skin allergy is called atopic dermatitis. You have a very common problem in human, and also there are dog breeds with severe problems with atopic dermatitis. And you may want to you should read a paper that we published recently. Let me see if I can send to everyone in meeting. You can read the paper by Tengvall et al. In Communications Biology, where we have identified to 2020, was it last year? Or was it this year? I think it was 22. Last year. Okay, so this paper is a very important contribution to the genetic underlying increased risk of developing atopic dermatitis. So I recommend, if you're interested, you can read that paper. Mm. But ethical considerations are extremely important in Sweden. I would actually say that it is more strict in Sweden than in the EU, the European Union. and it's probably definitely a lot more strict than you have in many other countries in the world. So we are the advocates for not doing research leading to suffering of the animals that we're using.
Okay. Yeah. Okay, thank you, Prof. Goran. Maybe I think it's a very interesting topic and discussion. A lot of inspiration for us, of course, to get what is inside, yeah, new idea, and also for the next maybe research and also collaboration. Okay, so
thank you very much.
yeah. Before maybe we give the appreciation, maybe from Prof. Roni, maybe Prof. Roni can say something maybe to Prof. Goran. Please, Prof.
Hello, Goran. Just want to thank you.
Hello, thank you
for your very nice presentation. So you open our mind. So
thank you very much, Roni.
we hope that in the future we can meet and then write kind of proposal, and we can do research together. Thank you very much.
I'm looking forward to that.
Thank you.
Who should I contact regarding this international program that I was invited to? Maybe Hikmayani can help me with that, but I was invited by 2 different persons apparently, so I'm not sure how to progress in that one.
I don't know. I try to find the person who invite you. So
well, I'm not sure which one of 2 different persons that I should.
Okay.
But I can ask Hikmayani again, because I was in contact with her with this problem.
Okay.
It's not a problem, but I don't want to. I should contact the right person, basically.
Okay.
Okay. Thank you.
Okay, thank you, Prof. Roni, and maybe we can also search for who is the contact, because now we have like a vice rector for connectivitas global, maybe like international, related with internationalization. Maybe so we will ask for that, maybe, and also Prof. Roni, of course.
You can send me an email of what is needed and when is the deadline or something like that. Okay, thank you very much.
Thank you. Thank you, Prof. Goran. And before we left, maybe we have some kind of appreciation of the certificates. Yeah, I would like to express thank you for the nice talk today, and an appreciation for your time and to be here. Allow me to hand over the certificate from the faculty of animal science. So on behalf of the dean, also head of department, while just a piece of the paper, and please accept this certificate, Prof. Goran.
Thank you very much.
As a speaker in joint webinar. Supervisor International 2023 for doctoral program IPB University. Please accept, Professor Goran.
Thank you very much. I appreciate this very much. Thank you very much.
Thank you for nice talk.
Okay, have a good evening.
Yes, thank you very
much.
much.
Bye.
Bye-bye.
Thank
you, Prof. Goran. Thank you, Prof. Roni.
Thank you. Makasih, Pak Gunawan.
Makasih. Ya, sama-sama, Bu.
Thank you.
All right, ladies and gentlemen. First, I would like to say thank you to Professor Asep Gunawan for leading the second webinar session. And as a thank you, I would like to give Professor Asep an appreciation in the form of certificate to the committee. Please share the certificate.
Thank you, Dava and others.
Yeah, thank you, Professor Asep, for leading the second webinar session. As a moderator, we hope that the knowledge that we get today can be used for good deeds.
Thank you.
Thank you, Professor Asep. All right, ladies and gentlemen. Nadia, what do we have next?
Okay, before we move on to our next agenda, I would like to remind all the participants, please fill out the presentation. Yes, you can access the website. Maybe the committee will send it on the chat box. Okay, so now we move on to our last agenda of today, a winner announcement. So yeah, congratulations for all the winner. So for those who win the prize, please kindly contact the contact person, the committee, or the committee also will contact you. So congratulations. Okay, everyone. Thank you to faculty, lecturers, guests, and participants for your participation today in this joint webinar CO-SUPERVISOR International 2023. We truly appreciate your participation of this program, and we look forward to you again on your other meeting.
That said, allow me to close today's webinar. I apologize for any mishaps that might have happened during the session, and we hope to see you again. Thank you and goodbye.
Goodbye. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
warahmatullahi wabarakatuh.
Kasih.
Makasih
Musik,
semuanya.
Mas. Musik.
Terima kasih Bapak, izin lab.
Iya.
Terima kasih Bapak Ibu, ini kolega dari Unsrat, dari USK, dari BRIN, dari semuanya nih, dari Jambi dan juga dari Kementan ya. Terima kasih semuanya Bapak Ibu, sampai jumpa lagi.
Sama-sama, Pak. Terima kasih. Oh.